Somewhere in your family tree, stretching back further than language, further than fire, further than anything you could reasonably call civilization, there is a moment when the entire human lineage nearly disappeared. Not metaphorically, not as a figure of speech, but in the cold, measurable language of genetics. Something happened roughly 1 million years ago that reduced the ancestors of every living person on Earth to a population so small, so fragile, so exposed to the random cruelties of chance, that by any reasonable accounting we should not be here. The number that keeps surfacing in the scientific literature is 1,280. Breeding individuals, not mouths, not bodies scattered across a continent, but reproductively active members of a population that would go on to eventually become you, your family, every human being alive today, every human being who has ever lived in the last several 100,000 years. Spread across an entire planet, 1,280 is not a town, it is barely a neighborhood. It is a number so small that a single bad season, a single catastrophic drought, a single wave of disease moving through the wrong valley at the wrong time could have ended everything. And yet here we are. That is the mystery. That is what keeps researchers up at night, what sends geneticists back to their models and paleontologists back to their field notes. What makes this particular chapter of deep human history feel less like ancient trivia and more like something urgent, something unresolved, something that still does not have a clean and satisfying answer? Before we go any further, it helps to understand what scientists mean when they use the word bottleneck, because it does not mean what people sometimes assume. A bottleneck is not an extinction event. It is not a moment where nearly everything dies in a sudden catastrophe, the way you might picture an asteroid impact or a supervolcanic eruption blotting out the sky. A bottleneck in the genetic sense is a severe and prolonged reduction in population size, one that lasts long enough to leave a measurable imprint on the DNA of every organism descended from those survivors. Think of it like squeezing a wide river through a narrow pipe. Whatever diversity existed before the squeeze, whatever genetic variation had accumulated across thousands of generations, gets dramatically compressed. Some of it is lost forever, simply by chance, because the individuals who carried certain genetic variants did not make it through, or did not reproduce, or happened to live in the wrong place. What emerges on the other side of the bottleneck is a population shaped by that narrowing, carrying the genetic signature of how few they once were. An extinction event ends a lineage, a bottleneck bends it, and what the evidence suggests happened somewhere between roughly 1.2 million and 800,000 years ago is one of the most severe bendings in the entire history of our genus. A squeeze so tight that it has left traces in human DNA that researchers are only now developing the tools to read clearly. That is the other thing worth holding on to as we move through this story. The signal in the genetics is strong. Multiple independent analyses, using different methodologies and different data sets, converge on the same general picture. A dramatic reduction in ancestral population size during a specific window of deep time. That part, the fact that something happened, seems increasingly difficult to dispute. What remains genuinely contested, what remains a legitimate and fascinating open question, is the cause. Was it climate? Was it a specific catastrophe? Was it isolation, competition, a slow grinding pressure from a world that had become more hostile in ways we cannot fully reconstruct? Was it some combination of all of these things, interacting in ways that are now impossible to fully separate? Science does not yet have a consensus answer, and anyone who tells you otherwise is getting ahead of the evidence. What makes this bottleneck particularly haunting is not just the number itself, but the duration. This was not a brief crash followed by a rapid recovery. Current estimates suggest the population may have remained at severely reduced levels for something in the range of 100,000 years. 100,000 years. To put that in perspective, modern Homo sapiens have existed as a recognizable species for somewhere between 200,000 and 300,000 years. The bottleneck we are talking about lasted, potentially, half as long as our entire species has walked the earth. Generation after generation after generation, born into a world where your kind was genuinely precariously rare. How do you survive that? How does any species thread a needle that narrow for that long without simply winking out? Early humans at this period were not who we are now. They lacked the cognitive complexity, the language, the social networks, the technological repertoire that modern humans use to buffer ourselves against uncertainty. They were intelligent, certainly, capable of planning and cooperation and the use of tools. But they were also vulnerable in ways that are hard to fully imagine, subject to predation, dependent on specific landscapes and food sources, without the global reach or the accumulated cultural knowledge that eventually made our lineage so extraordinarily hard to kill. And yet something worked. Something about those 1,280 individuals, or some fluctuating group in that general range, something about where they were or how they organized themselves, or how they responded to whatever pressure was bearing down on them, was enough. Barely enough, perhaps. Enough by the narrowest of margins, but enough. That is what this story is really about. Not just the crash itself, but the survival that followed it. Not just the question of what caused the bottleneck, but the deeper question of what it might have done to us. Because there is a reasonable scientific argument that events like this do not simply reduce a population and leave everything else unchanged. They can reshape the genetic landscape of a species, eliminating certain variations, concentrating others, potentially accelerating certain kinds of change. They can act, in a sense, as an involuntary experiment in what a species is made of at its most reduced and most essential. We are, all of us, the descendants of whoever made it through. Whatever qualities, whatever capacities, whatever combinations of trait and circumstance allowed those individuals to survive and reproduce during one of the most precarious periods in our lineage's history, those are the qualities that got passed forward. They are woven into us, literally, at the level of the genome, which means understanding the bottleneck is not purely an exercise in ancient history. It is, in a strange and somewhat vertiginous way, a form of self-knowledge. Over the course of this video, we're going to move through what is currently known, what is actively debated, and what remains genuinely mysterious about this period. We will look at how scientists extract population signals from ancient DNA, what the fossil and archaeological records can and cannot tell us, how the climate of that particular window in Earth's history might factor into the picture, and what serious researchers are currently arguing about the possible causes. We will be careful about certainty where certainty is not warranted, and honest about the places where the evidence still leaves enormous room for interpretation. But before we go any further, a quick pause. We are genuinely curious about who is watching this right now, and from where. It is always a bit remarkable to think that someone might be sitting with this video in the middle of the afternoon in one part of the world while someone else is watching it in the early hours of the morning somewhere else entirely, all drawn to the same question about something that happened 1,000,000 years ago. So if you feel like it, leave a comment. Tell us where you are in the world right now and what time it is there. There is something fitting about that. A species that survived a near erasure now scattered across the entire planet, watching videos about our own deep past from thousands of different locations simultaneously. Take a moment if you want, and then let us go back, a very long way back. So let us slow down for a moment and look more carefully at that number, because it is doing a lot of work in this story, and the way it gets reported in headlines and shared across the internet tends to flatten something genuinely subtle into something that sounds more dramatic and more precise than it actually is. The 1,280 figure does not refer to a head count. It is not saying that at some particular moment, on some particular afternoon a million years ago, there were exactly 1,280 human ancestors alive on the planet. That framing, which is understandably tempting because it is vivid and concrete, misrepresents what the number actually measures. What researchers are talking about when they use a figure like this is something called effective population size, and understanding the difference between effective population size and raw head count is the key to understanding what the bottleneck evidence is and is not actually claiming. Effective population size, often written as NE in the scientific literature, is a statistical concept that describes the size of an idealized population that would produce the same amount of genetic drift as the real population being studied. That requires a bit of unpacking. Genetic drift is the random fluctuation in gene frequencies that happens simply because reproduction is not a perfectly even process. Some individuals have more offspring than others, not because they are necessarily better adapted, but because of chance. Over time, drift erodes genetic diversity in a predictable way, and the smaller the population, the faster diversity erodes. So when geneticists estimate an effective population size, they are essentially asking, given the pattern of genetic diversity we observe in this lineage today, what population size would have produced that pattern? It is a way of reading the past through the statistical fingerprints left in DNA. Here is where it gets important. The effective population size of a species is almost always smaller than its actual census population, sometimes dramatically so. There are several reasons for this. If a population is structured into isolated groups that do not interbreed freely, the effective size can be much lower than the total head count would suggest, because the genetic drift happening in each isolated group is not being averaged out across the whole. If some individuals contribute far more offspring than others because of social structure, because of geography, because of unequal access to resources, the effective size shrinks relative to census size for the same reason. Fluctuations in population size over time are also heavily weighted toward the smaller end, because a brief crash leaves a disproportionately large signature in the genetics compared to extended periods of abundance. What this means practically is that an effective population size of 1,280 could be consistent with a real-world population somewhat larger than that, perhaps significantly larger, depending on the degree of structure and inequality in who was actually reproducing. Some researchers have suggested the total census population during the bottleneck might have been several times the INI estimate. Others think the two figures were probably reasonably close. The honest answer is that separating them with confidence across a gap of nearly 1 million years is genuinely difficult. The other thing the headline framing tends to obscure is what a long bottleneck looks like in the data versus a short one. A brief population crash, the kind that might happen in a single catastrophic generation, leaves a very different genetic signature than a sustained reduction spread across tens of thousands of years. What the current research appears to be picking up is not a cliff edge, a sudden plunge to a small number followed by immediate recovery. It looks more like a long, slow trough, a period during which the ancestral population remained small and genetically constrained for an extended stretch of time. That is, in some ways, a stranger and harder to explain signal than a single dramatic catastrophe would be. A disaster you can name, a century after century of sustained smallness, is a different kind of puzzle. It is also worth being honest about the precision implied by a number like 1,280. The decimal point specificity of it can create a false sense of certainty. Different modelling approaches, different assumptions built into the statistical frameworks, different ways of calibrating the molecular clock used to date these events can produce somewhat different estimates. The 1,280 figure comes from a specific study using a specific methodology, And while other independent analyses have converged on broadly similar conclusions about a severe and prolonged reduction in this general time window, the exact number should be held loosely. What matters is the order of magnitude. We're not talking about tens of thousands. We're talking about something in the low thousands, possibly lower, sustained across what may have been a geological eye blink, but felt, from the inside of a human life, like an eternity. So what kind of pressure could push me down that far for that long? That is the real question, and it is the one that drives everything else in this story. A brief drought might crash a population for a generation, and then allow recovery. A single volcanic winter might do the same. But to keep a lineage bottlenecked across potentially 100,000 years requires something more persistent, more structural, more woven into the conditions of life during that period. It suggests not a single event but a sustained state of things, a world that was, for reasons still being debated, genuinely hostile to the proliferation of our ancestors. Whether that hostility came from the sky, from the landscape, from competitors, from the deep structure of the population itself, or from some combination of all of these pressures stacking on top of each other, is exactly what researchers are still working to understand. And to begin making sense of any of it, we first need to understand how scientists can see that far back at all. So how does any of this work? How do you look at a person living today, sequence their genome, and come away with a confident claim about what was happening to their ancestors nearly 1 million years before anyone thought to write anything down? It sounds almost like a parlor trick, but the method behind it is genuinely elegant, and understanding it, at least roughly, is essential for grasping both what the bottleneck evidence is actually saying and why reasonable scientists can still argue about what it means. Start with a thought experiment. Imagine tracing your family tree backward. You have 2 parents, 4 grandparents, 8 great-grandparents, and so on. Go back far enough and the tree fans out so rapidly that the number of ancestors you theoretically have exceeds the number of humans who were alive at the time. That means something important. The tree must collapse back on itself. People who appear in multiple branches of your ancestry were, at various points, the same individual. The further back you go, the more the branching tree begins to converge into a smaller and smaller number of actual people. Geneticists call this process coalescence, and it is the conceptual heart of the method. Now apply this not to your whole family tree, but to a single stretch of DNA. Any given segment of your genome has a history. It was copied from someone, who copied it from someone, who copied it from someone before them. Trace that lineage backward, and you eventually reach a single ancestral copy, one individual in the deep past from whom all present-day copies of that segment descend. That point is called the coalescent, and the time it takes to reach it, averaged across many different segments across the genome, tells you something about how big the ancestral population was. Here is the key insight. In a large population, 2 randomly chosen copies of the same DNA segment could have come from any of a huge number of ancestors, so on average you have to go very far back before you find the common ancestor they share. The coalescent time is long. In a small population there are fewer ancestors to choose from, so 2 copies are more likely to share a recent common ancestor. The coalescent time is short. When geneticists observe that a particular stretch of DNA across many living individuals all traces back to a common ancestor within a relatively tight window of time, they are seeing the fingerprint of a population that was small during that period. The smaller and longer the bottleneck, the more of the genome will show this compressed pattern of coalescence. But knowing when coalescence happened requires a clock, and that clock is built from mutations. Every time DNA is copied, there is a small chance of an error, a substitution of one letter in the sequence for another. These errors accumulate at a roughly predictable rate. By counting how many mutations separate 2 lineages and dividing by the estimated rate at which mutations accumulate, you can estimate how long ago those lineages diverged. This is what people mean when they talk about the molecular clock. It is not perfectly precise, and calibrating it accurately is one of the persistent technical challenges in this field, but it provides a framework for dating genetic events that would otherwise be inaccessible. The mutation rate itself has been a source of genuine debate. Early estimates came from comparing human and chimpanzee genomes and dividing by an assumed divergence time based on fossil evidence. More recently, researchers have been able to measure the mutation rate more directly by sequencing parents and their children and counting new mutations in each generation. These two approaches have given somewhat different answers, and the difference matters when you are projecting estimates back nearly 1 million years. A clock running slightly slow or slightly fast compounds into significant error across that kind of timescale. This is one of the reasons why the specific number attached to the bottleneck, that 1280, should be held with some flexibility. Change your assumptions about the mutation rate, and the estimated timing and severity of events in the deep past shift accordingly. There is another layer of refinement that has made modern genetic inference considerably more powerful than earlier methods, and it has to do with recombination. Chromosomes are not inherited as single, unbroken units from parent to child. Instead, during the formation of reproductive cells, paired chromosomes exchange segments with each other in a process called recombination. This shuffling means that a single chromosome you carry today is actually a patchwork of segments with different ancestral histories. The stretch near one end might trace back to one ancestral population, while a stretch near the other end traces back through a completely different lineage. This turns out to be enormously useful, because different segments of the genome are essentially independent witnesses to the same deep history. Researchers can treat each segment as a separate data point. Instead of having one family tree to analyze, you have thousands of overlapping trees, each one offering a slightly different angle on the same past. Statistical methods that pool these signals, essentially asking what population history is most consistent with all of these different trees simultaneously, are far more powerful than anything based on a single genetic locus. The more of the genome you can read, and the more individuals you can include in the analysis, the sharper the picture becomes. One of the slightly counterintuitive aspects of this kind of inference is that ancient events can sometimes be easier to detect than recent ones, at least in certain respects. A population crash that happened 50,000 years ago might be partially obscured by subsequent demographic changes that have had time to overwrite parts of the genetic signal. But an event that compressed genetic diversity nearly 1 million years ago left its mark on the very deep structure of human genomic variation, the long branches of the ancestral trees, the patterns of similarity and difference that underlie everything that came after. Subsequent expansions and migrations and mixings have been layered on top, but they cannot fully erase the deep compression that occurred so far back. It is there in the base of the trees, visible to methods that know how to look for it. That said, the limitations of the approach are real and worth naming clearly. Every statistical model of population history is, by necessity, a simplification. The actual history of a species is messier than any model can capture. Populations are not panmictic blobs that drift together uniformly. They are structured into groups, some of which interbreed more with each other than with distant groups, some of which go locally extinct while others expand, some of which experience very different selective pressures on different parts of their genome. Models that do not adequately account for this structure can produce misleading estimates of effective population size. What looks in the data like a global bottleneck might, under some models, be partially explained by a particular kind of subdivision among subpopulations. Conversely, structure that the model does not capture might cause it to underestimate the severity of a real bottleneck. There is also the problem of what statisticians call model misspecification. The mathematical frameworks used to infer population history make assumptions about how populations change over time, how selection works, how recombination and mutation proceed. If those assumptions do not match reality well enough, the inferences they produce can be systematically wrong in ways that are difficult to detect from inside the model itself. Researchers try to guard against this by using multiple independent methods and checking whether they converge on similar conclusions, and to a meaningful degree the different analyses looking at the deep human bottleneck have converged. But convergence between models does not guarantee truth if the models share underlying assumptions that are themselves flawed. What this means for interpreting the bottleneck evidence is something like this. The broad signal, a severe and sustained reduction in the effective population size of our ancestors somewhere in the region of 900,000 to 800,000 years ago, is robust enough that it has survived multiple independent methodological approaches. The order of magnitude of the compression appears to be real. But the specific numbers, the precise timing, the exact duration, the relationship between effective population size and actual census population, these are all estimates with real uncertainty attached to them. They are the best answers science currently has, not the final answers. And it is worth sitting with that distinction for a moment, because what the genomic evidence is really doing is something remarkable even in its imperfect form. It is letting us hear an echo from nearly 1 million years ago, a compression in the genetic variation of living humans, a statistical whisper passed down through countless generations, is telling us that our ancestors came terrifyingly close to vanishing from the world. The signal is real. What we do not yet fully understand is the story behind it, the particular collision of forces and circumstances that squeezed the thread of our lineage so thin for so long. And that is where the science gets even more interesting. For a long time, that echo from the deep past was more of a vague murmur than a sharp sentence. Genetic studies going back to the early 2000s had hinted at ancient fluctuations in population size, including possible contractions. But the resolution was fuzzy, the timescales broad and the models relatively simple. You could see that something had happened, but not quite when, not quite how severely, and not quite how long it had lasted. That changed when a new generation of methods, and one paper in particular, pushed the technique to its limit. The core idea behind these recent high-profile analyses is conceptually straightforward, even if the math is not. Take whole genomes from living humans. Slice those genomes up into many small segments. For each segment, reconstruct a tiny family tree, who seems to share a recent ancestor with whom, how deep those branches go, where they split and where they converge. Then pool all those tiny trees together and ask, what history of population size through time would most naturally produce a forest of genealogies that looks like this? In practice, that means turning the logic of coalescence into a time machine of sorts. Instead of assuming a particular population history and simulating what patterns of genetic variation it would produce, these methods go the other way around. They start from the patterns actually observed in the genomes and work backward, searching for the most plausible trajectory of population size that could have given rise to those patterns. The technical tools with the unwieldy names, methods like PSMC, MSMC, SMC++, and more recently, approaches that treat the genome as a vast set of correlated genealogies, are all variations on this theme. They differ in how they model recombination, how they approximate the underlying coalescent process, how many genomes they can handle simultaneously, and how they navigate the impossible task of exploring every conceivable demographic history. But the shared ambition is the same, to infer a curve of effective population size over time, not just a single number. That phrase, inferring population size through time, sounds simple enough. In practice, it means that for each point in the distant past, say 200,000 years ago, 500,000 years ago, 900,000 years ago, the method produces an estimate of how large the ancestral effective population was at that time, along with a sense of how confident we should be in that estimate. Stitch those points together and you get a population history curve, rising where the ancestors expanded, falling where they went through lean times, leveling off where things were relatively stable. The recent bottleneck study that stirred so much debate essentially produced such a curve and found something dramatic. When the authors ran their model on whole genome data from hundreds of present-day people from around the world, the inferred population size did not simply wobble up and down. It plunged. According to their reconstruction, somewhere around 930,000 years ago, the effective size of the ancestral population leading to modern humans began to collapse from a relatively healthy tens of thousands down to a figure on the order of 1,000, and it stayed low. Depending on the exact mutation rate and generation time assumptions you plug into the model, the window of this compression stretches from roughly 930,000 to about 810,000 years ago, something on the order of 100,000 years of sustained small size. That is the prolonged low E you may see referenced in discussions of this work, NE being shorthand for effective population size, not the census head count, not the number of actual individuals walking around on the landscape, but the number of breeding individuals in an idealized population that would produce the same rate of genetic drift, the same pace of random change in gene frequencies that we infer from the data. Under this reconstruction, NE bottoms out at around 1,280, with some uncertainty in both directions. The authors emphasize that this low effective size persists for many thousands of generations, rather than being a brief blip. To a geneticist, that duration matters as much as the depth of the minimum, because a short, sharp crash can leave a different imprint on the genome than a long plateau at reduced size. A prolonged low-E steadily erodes variation, allowing random chance to sculpt the genome much more strongly than selection, and increases the odds that rare, slightly harmful variants may stick around simply because there are too few lineages for natural selection to efficiently remove them. It is worth being very clear about what is actually measured here and what is being inferred. The sequences themselves, the A, C, G, and T letters that make up each genome, are measured directly. So are the patterns of similarity and difference between individuals, where they share long stretches of nearly identical DNA, where they differ at scattered positions, how those differences correlate across the genome. From those raw patterns, the model infers a set of underlying genealogies, approximate branching histories for different segments of the genome. Those genealogies are not observed in the same way a bone in a cave is observed. They are statistical constructs, best fit trees that explain the data under a particular set of assumptions about recombination, mutation, and inheritance. From the genealogies, the model then infers the distribution of coalescent times, the ages at which lineages merge back into common ancestors, and how that distribution changes with depth. And from that distribution, using the mathematics of coalescent theory, it infers the effective population size through time, because the rate at which lineages coalesce is tightly linked to how many ancestors were available at different moments in the past. Every step beyond the raw sequences themselves is a layer of modelling. The genealogies are inferred. The timing of their branching is inferred using a mutation clock that itself has to be calibrated. The translation from coalescent rates to NE assumes a particular kind of population structure, often something close to a single, well-mixed population. None of this makes the results meaningless. It simply defines the chain of reasoning and highlights where uncertainties can creep in. So why did this particular analysis, among many attempts to reconstruct human demographic history, grab so many headlines and provoke so many responses from other scientists? Part of it was the sheer depth of the window the authors claimed to be peering into. Most demographic reconstructions of humans focus on the last few 100,000 years, periods populated by Neanderthals, Denisovans, and anatomically modern Homo sapiens, where fossil and archaeological evidence can at least partly cross-check the genetic picture. This study reached well beyond that, out toward 900,000 years ago, into a time when the fossil record is far thinner and more ambiguous. The idea that the genomes of people alive today could still carry detailed information from the era was, by itself, attention-grabbing. Another part was the duration and severity of the inferred bottleneck. A momentary dip in knee can often be folded into existing narratives without much trouble, but a 100,000-year stretch with an effective size hovering around 1,000 or so is not a small tweak to the story of our origins. It is a major plot point. It implies that for a vast span of time, our lineage was skating on the edge of viability, its genetic diversity shrinking, its evolutionary trajectory constrained. And then there were the implications. A bottleneck of that severity and duration, occurring in that particular window of time, lands very close to periods when other lines of evidence suggest significant changes in hominin evolution. Around the same broad era, there are hints of transitions in brain size, shifts in stone tool technologies, and rearrangements in the distribution of early Homo species across Africa and Eurasia. The temptation to connect these dots, to suggest that a demographic crisis may have been linked to, or even helped drive, some of these evolutionary shifts, is strong. The authors themselves gestured cautiously in that direction, noting that such a prolonged constriction could, in principle, have facilitated rapid genetic change by amplifying the effects of drift and selection on a now tiny population. All of that combined, the ambitious methodology, the deep temporal reach, the stark population minimum, the suggestive alignment with other evolutionary milestones, made this study difficult to ignore. Its conclusions were bold enough that they demanded scrutiny. Critics questioned the assumptions about population structure, the robustness of the mutation rate calibration, the sensitivity of the inferred bottleneck to modelling choices. Supporters pointed to the convergence with other, older methods that had also suggested a substantial population contraction in roughly the same window, albeit with less dramatic numbers. What emerged from the debate was not consensus about every detail, but a sharpened sense of what was at stake. If the broad outline of this reconstructed bottleneck is even roughly correct, it forces us to rethink the backdrop against which key chapters of human evolution unfolded. It suggests that our lineage did not drift steadily upward in numbers, slowly spreading and diversifying, but instead passed through a drawn-out period of extreme vulnerability nearly 1 million years ago. And that raises the next, unavoidable question. If our ancestors really were reduced to such a small, precarious population for so long, what exactly was happening in the world around them? What forces in the environment, the climate, the landscape of predators and competitors conspired to hold them down on that narrow ledge for 100,000 years, and what finally let them climb back up? Once you start asking what was happening in the world around our ancestors, you run straight into an uncomfortable fact. You're no longer just reading off a simple picture from the genomes. You're balancing on a scaffolding of assumptions. One of the most important pieces of that scaffolding is something called the mutation rate. Every time DNA is copied, a tiny fraction of letters change. Accidents of chemistry and biology that usually go unnoticed, but that, over many generations, act like ticks on a clock. The longer two lineages have been separated, the more of these little differences build up between them. To turn this much difference into this many years, you have to decide how fast the clock is ticking. The catch is that we do not have a single, universally agreed upon tick rate for the human mutation clock deep in time. We have estimates, and those estimates depend on how you measure. One way is to compare humans and chimpanzees, count how many differences separate us, and then divide by how long ago we think our lineages split, based on the fossil record. That gives you a relatively fast mutation rate. Plug that faster rate into a demographic model, and all the events it reconstructs get pulled closer to the present, because you accumulate the observed differences in less time. Another way is to use pedigrees, actual families where you can sequence parents and children and directly count how many new mutations appear each generation. Those studies tend to produce a slower mutation rate. Slower ticks mean you need more years to build up the same pile of differences, so the same pattern of genetic variation now maps onto older dates. The difference between these clocks is not a mere rounding error. Depending on which numbers you use, a demographic event that one researcher would place at 900,000 years ago might shift to 600,000 years ago in another reconstruction, or stretch outward past 1,000,000 years. The shape of the bottleneck signal in terms of earlier versus later generations may be reasonably constrained, but its absolute position on the calendar is remarkably flexible. Closely related to the mutation rate is another quiet but powerful assumption: generation time, the average span between birth and reproduction. In modern humans, people often use something in the range of 25 to 30 years. But there is no guarantee that the hominins alive 800,000 or 1,000,000 years ago reproduced on the same schedule as we do. If early members of our lineage tended to have children younger, say every 20 years on average, then 5,000 generations worth of genetic change would represent a shorter span of calendar time than if they reproduced every 30 years. The demographic models work in units of generations. The genomes do not know how long a generation lasted. When we say the bottleneck lasted around 100,000 years, what we really mean is it lasted some number of generations that, given our assumptions, translates into about 100,000 years. Change the assumption and the translation changes. Mutation rates and generation times are intertwined in an almost invisible way. You can slow down one and speed up the other and still fit the same genetic data, but the dates on the timeline will slide. That sliding is one reason why different studies, using similar methods on similar data, can disagree by hundreds of thousands of years about when exactly a particular contraction or expansion occurred. And that is before you even touch what may be the trickiest assumption of all: how the ancestral population was structured. Most coalescent-based reconstructions start from a simplifying picture. They imagine one big, more or less well-mixed population, where any individual is about as likely to mate with any other, and where lineages drift and coalesce within that single, continuous pool. Under that picture, a period when many genealogical branches merge quickly, when lots of lineages share common ancestors in a relatively short span of time, naturally maps onto a small effective population size. Fewer ancestors are available, so lineages bump into each other faster, but real populations are almost never that simple. They are messy and lumpy. They are divided into subgroups, by geography, by rivers and mountains, by shifting climate zones, by cultural boundaries that do not all interact equally. Individuals in one region may mostly mate among themselves, only occasionally exchanging migrants with neighbouring regions. Some subgroups may stay small and isolated for long periods, while others may be larger and more connected. That kind of structure by itself can create patterns in the genealogies that, to a model assuming a single panmictic population, look like changes in overall size. If two subpopulations that have been partly isolated for a long time suddenly start exchanging genes more freely, lineages that have been drifting separately can suddenly find many opportunities to coalesce. The model, unaware of the merging subgroups, will interpret that sudden burst of coalescences as a decline in any. It seems as if, all of a sudden, there were fewer ancestral individuals because lineages are converging faster. The reverse is also true. If one widespread population gradually fragments into smaller, more isolated pockets, lineages that used to coalesce readily now have a harder time finding common ancestors because they are confined within their own local pools. To a model that does not know about the fragmentation, that slowdown in coalescence can look like an expansion in effective size. In other words, changes in how populations are connected in space, merging, splitting, growing more or less isolated, can mimic the genetic signatures of crashes and booms, even if the total census number of hominins on the landscape did not change nearly as dramatically as the inferred curve suggests. This is not just a theoretical worry. Several follow-up studies, reacting to the dramatic bottleneck claim, have shown that if you simulate a world with multiple semi-isolated hominin groups, each with its own demographic quirks, and then you feed that synthetic genetic data into a model that assumes a single well-mixed population, you can generate an apparent prolonged low NE phase that sits roughly in the same time window as the published bottleneck. No asteroid impact, no supervolcano required, just a tangle of subpopulations separating and rejoining over time. Under that lens, the striking crash to 1,280 might not reflect a literal global collapse in the number of our ancestors. It could instead be an artifact of squeezing a complex structured reality into a simpler model. The model has only one knob, population size, to explain shifts in the rate at which lineages coalesce. So whenever the true history throws it something that changes coalescence rates for other reasons, like a reorganization of population structure, it reaches for the only knob it has and turns it. None of this means the bottleneck signal is purely illusory. The fact that multiple independent methods with different strengths and weaknesses have pointed to some kind of contraction in roughly this window suggests that something genuine may well be there. But something happened is a much weaker and more cautious statement than our ancestors were reduced to exactly 1,280 breeding individuals for 117,000 years, starting precisely 930,000 years ago. When you look closely, every piece of the timeline is bracketed by caveats. Shift the mutation rate, and the whole curve slides forward or backward in time. Tweak the average generation length and its duration stretches or compresses. Allow for structured populations instead of a single panmictic one, and the sharpness and depth of the trough may soften or even break up into a series of smaller dips and plateaus. What we are left with is a signal that many researchers take seriously, but few would claim to have fully pinned down. There does appear to be a period in the mid-Pleistocene when the lineages leading to modern humans passed through a phase of reduced genetic diversity and unusually frequent coalescences. Whether that phase reflects a dramatic species-wide crash, a drawn-out reconfiguration of scattered hominin groups, or some combination of both is still under active debate. And that uncertainty matters when we try to tie the genetic story to the rocks and bones. If the trough in Ni really centers around 930,000 years ago and lasts for 100,000 years, it overlaps one set of climatic and ecological changes. If, under a different clock and a more structured model, it shifts to 750,000 or 650,000 years ago, it lines up with a different cast of characters entirely. Other glacial cycles, other hominin fossils, other archaeological horizons. So before we can confidently say what kind of world squeezed our ancestors into that narrow passage, we have to decide which version of the passage we are talking about. The genomes are whispering that there was a constriction, but exactly when it tightened, how narrow it became, and what sort of maze of populations it wound through, those are questions that grow harder, not easier, the further back we push. To make progress, we have to turn away from the curves on a computer screen and look outward at the world they supposedly describe. What was actually happening on Earth between about 1.2 million and 800,000 years ago? What did the climate look like? The landscapes, the animal communities, the other hominins sharing the stage? Only by setting the uncertain genetic timeline against that shifting backdrop can we start to see which stories about our near erasure are even remotely plausible, and which ones crumble under their own assumptions. If you could zoom out from any one band of hominins walking across Africa or Eurasia 1 million years ago, and instead see the whole planet at once, the first thing that would strike you is how restless it all was. Ice sheets in the high latitudes were advancing and retreating like a slow, grinding tide. Coastlines shifted, deserts inhaled and exhaled, swallowing grasslands and then spitting them back out thousands of years later. Forest belts marched north and south, up and down mountains, tracking temperature and rainfall. The window between roughly 1.2 million and 800,000 years ago sits right on top of a turning point in that restlessness, a period geologists call the early Middle Pleistocene transition. Hidden in stacks of seafloor mud and polar ice is a record of how Earth's climate rhythm changed, and with it, the rules that governed where plants, animals, and our ancestors could survive. For hundreds of thousands of years before this transition, the planet's ice ages followed a relatively modest beat. Glacial and interglacial cycles were paced mainly by a 41,000-year wobble in Earth's tilt, and the differences between cold and warm phases, while serious, were not as extreme as what would come later. Think of it as a metronome ticking steadily. Ice sheets grow, seas fall, climates cool, then the tilt shifts, the ice retreats, forests and grasslands surge back. Around a little over 1 million years ago, that metronome started misbehaving. The periodic nudges from Earth's orbit and tilt were the same, but the climate's response began to reorganize. Over the span of a few 100,000 years, the dominant rhythm of the ice ages stretched from 41,000-year cycles to longer, more irregular swings on the order of 100,000 years. Glacials became deeper, longer, and more punishing. Interglacials, in turn, sometimes spiked to surprisingly warm conditions. This shift is often called the mid-Pleistocene transition, and although the exact mechanisms are still debated, changes in ice sheet dynamics, carbon dioxide feedbacks, erosion of continental shelves, and more, it marks a world where variability itself was ramping up. Instead of milder, more frequent swings, you get fewer, harsher ones. The amplitude of the climate roller coaster grew. For hominins, that mattered more than the abstract physics of orbital cycles. What they would have experienced were the consequences. Where water was, where plants grew, where animals migrated, which familiar territories turned suddenly hostile. During colder glacial phases, vast amounts of water locked up in ice lowered sea levels, sometimes by more than 100 meters. Shelves that are underwater today became broad coastal plains. Land bridges emerged between islands and continents, opening migration routes and then drowning them again when the ice melted. Rain belts shifted toward and away from the equator. In Africa, these swings rearranged the balance between forest, woodland, and open savanna. In Eurasia, they repeatedly pushed temperate ecosystems southward and then allowed them to creep back north. These were not gentle, uniform adjustments. They were patchy. A region that had been a reliable mosaic of woodland and grassland might, over the course of a few glacial cycles, flip into more open, arid steppe and then back again. Lakes expanded and contracted. River courses shifted or dried. What had been a green highway of resources for hominins and other animals could fragment into isolated oases separated by stretches of marginal land. That is what habitat fragmentation meant on the ground. Forests did not simply vanish. They broke into islands of canopy in a sea of grass or scrub. Grasslands did not endlessly sprawl. They narrowed into corridors along rivers or plateaus. Mountains, already barriers, became even more decisive dividers as climate zones climbed or descended their slopes. For a creature that walked, hunted, gathered, and carried everything it needed, this mattered at a very practical level. Where can you find water at the end of the dry season? Where do the herds go when the rains fail in one basin, but fall in another? Which valley that your grandparents knew as a dependable refuge is now a treeless, wind-scoured plain? As glacial-interglacial cycles stretched and intensified, these questions stopped having stable answers. A group of hominins might adapt to a particular pattern, a mixture of woodland fruit, tubers and gallery forests, antelope on the open ground, only to watch that pattern unravel over the course of a few thousand years. A riverine corridor linking two regions might pinch off into a chain of dwindling wetlands. A forest belt that once connected Central and Eastern Africa could retract and fragment, isolating populations in separate refugees. From the perspective of population genetics, this is exactly the kind of ecological backdrop that encourages splitting and isolation. As habitats fragment, so do the populations living in them. Groups that once exchanged mates along continuous bands of usable land now find themselves cut off by stretches of desert, by barren highlands, by forests too dense and unproductive for their foraging style, or by grasslands too exposed and dry. Gene flow slows or stops. Local cultures and adaptations proliferate in pockets. Then, when the climate swings back, when glaciers retreat, rainfall patterns shift again, and corridors reopen. Those once isolated groups may come back into contact, carrying slightly different genes, different behaviours, and different pathogens, mixing resumes but not as if nothing had happened. The genetic record of that separation and reunion persists. This dynamic is not unique to hominins. Large herbivores, carnivores, and many smaller species would have ridden the same waves of expansion, contraction, and isolation. But for a lineage that depended on relatively large home ranges, flexible diets, and the ability to move across landscapes, the consequences were especially stark. Food reliability was no longer something you could assume based on inherited knowledge alone. It was a gamble against a shifting climate. Periods of particularly harsh glacials with prolonged aridity in key regions may have thinned populations, pushing them into the best remaining refuges, wetter highlands, lake margins, coastal zones, or forest pockets that held onto moisture. In those refuges, populations may have been smaller and more crowded, facing intensified competition for the same edible plants and prey, and more frequent encounters with other predators drawn to the same shrinking resource bases. Conversely, some interglacials might have been times of opportunity, when expanding forests or grasslands opened new routes out of Africa or into underused parts of Eurasia. Migration pressure would have oscillated with the climate. When local conditions deteriorated, moving became not just tempting but necessary. When new lands became habitable, exploring them offered an escape from crowding or scarcity. From a distance, all of this looks like a recipe for environmental stress, not one single dramatic catastrophe, but an accumulation of pressures, more volatile climate swings, more fragmented habitats, more frequent reorganizations of where life could thrive. It is easy to imagine how, against this background, certain regions might see long-term declines in hominin numbers, punctuated by bursts of movement and mixing when conditions briefly improved. It is also easy to oversimplify. The mid-Pleistocene climate transition did not create a uniformly hostile world. Some areas may have become more productive or more stable. Different hominin species and populations would have been affected in different ways, depending on their ranges, their diets, and their technological abilities. What was a crisis for one group might have been a window of expansion for another. That nuance matters when we zoom back in on the genetic bottleneck debate. The timing of the most severe glacial phases in this window, the reorganization of climate cycles, and the evidence for repeated habitat fragmentation all line up well enough with the broad mid-Pleistocene constriction hinted at in our genomes to make environmental stress a plausible contributor. A world of shrinking refuges, unstable food webs, and disrupted migration routes is exactly the kind of arena where populations could dwindle and genetic diversity could be lost. But plausible is not the same as proven. There is no single glacial, no single drought, no single climate event in this 1.2 to 0.8 million year span that obviously screams, this is it, this is when humanity almost vanished. Instead, what we see is an Earth system gradually shifting into a rougher mode of operation, and a hominin world forced to track that shift with its feet, its tools, and its social structures. If a bottleneck or a prolonged phase of low effective population size really did shape our lineage in this era, it was likely not the fingerprint of one clean, isolated blow. It would have been carved by the repeated grinding stress of living in a world whose basic rhythms were becoming more extreme and less predictable. To sort out how much of the genetic signal can be laid at the feet of this changing climate, we have to turn to the other witnesses from that time, the scattered bones of hominins and the stone tools they left behind. Those fossils and artifacts are sparse and often ambiguous, but they are our best chance at seeing who was actually on the landscape during these climatic upheavals, and whether their story really looks like one of near erasure. If you spread out a world map and mark every place where fossils of our genus have been found from this window, say, between about 1.2 million and 800,000 years ago, you don't get a dense cloud of points. You get a handful of scattered dots, a skull fragment here, a jaw there, teeth, bits of limb bone, a partial cranium if you're lucky. Many of them belong to what paleoanthropologists broadly file under Homo erectus and its close relatives, long-legged, fully upright walkers with brains larger than earlier hominins but smaller than ours, and with bodies built for covering ground. In Africa, there are fossils some researchers would call early Homo heidelbergensis, or Homo rhodesiensis. Others might lump into late African erectus, and still others prefer to leave in a grey zone of archaic Homo. In Eurasia, you see classic Homo erectus in places like Java and China, and more fragmentary remains in Georgia and Europe, hinting at populations usually called Homo antecessor or early Homo heidelbergensis. The labels can make it sound as if there were a neat parade of species, one after another. The reality, especially in this time slice, was probably much messier. Look closely, and the fossils suggest a world where several related hominin forms may have coexisted, overlapped, and interacted across Africa and Eurasia. But before trying to read too much into that pattern, there's a hard lesson that paleontology keeps forcing on us. The fossil record is not the census. It's not even a decent sample. It's a biased, threadbare archive with most of its pages missing. Bones have to get buried quickly in the right conditions to fossilize at all. Then they have to survive scavengers, chemical decay, erosion, and the random violence of geology. Then, after all that, the right piece of land has to be exposed at the surface in the present, and someone has to happen to be looking in exactly the right place. Most individuals who ever lived left no trace we will ever find. Most populations, most sites, most regions are simply gone to us. Sparse fossils do not mean sparse hominins. 100 bones spread over 400,000 years could, in principle, represent millions upon millions of individuals whose remains never made it into the record. That's why you have to be careful when you see a timeline that shows hominin species as clean, continuous bars across a page. Those lines are often stitched between a few isolated points. Whole expansions, contractions, and local extinctions could be hiding in the gaps between the fossils we actually have. Even so, sparse does not mean useless. The fossils we do have carry clues about where our ancestors were, what they were dealing with, and what their bodies were adapted to in this turbulent slice of the Pleistocene. Take geographic coverage. Our fossil dots are not sprinkled uniformly across the continents. They cluster in very particular zones, parts of East and South Africa, a few river valleys and cave systems in Europe, particular basins in China, parts of Indonesia. Huge swaths of Africa have yielded almost nothing from this window. Most of Arabia, much of South Asia, big chunks of Southeast Asia and Central Eurasia are blank, not because hominins definitely weren't there, but because nobody has found them, or in many cases nobody has really looked under the right conditions. This is what researchers mean by geographic bias. Where we have fossils often tells us more about where the right rocks are exposed and where field work has been funded than about where hominins actually lived. Regions with deep sediments that have been uplifted and eroded just so, like parts of the East African Rift, become our main windows into the past, while other regions that may have been just as important for hominin history remain opaque. That bias makes it risky to say, for example, Homo erectus only lived here, or early Homo heidelbergensis first appears there. The first appearance of a fossil in the record is usually the first time conditions happen to line up for preservation and discovery, not necessarily the true birthplace of a lineage. Yet the geography of fossils still hints at interesting patterns. By 1 million years ago, we find Homo erectus-like remains not only in Africa, but across large parts of Eurasia, from Dmanisi in Georgia to sites in northern China and Indonesia. That distribution, combined with their long legs and narrower hips, paints a picture of hominins that were increasingly committed to life on the move, able to follow shifting habitats and animal herds over long distances. Morphology, the shape and proportions of bones and teeth, adds more pieces to the puzzle. Compared to earlier hominins, these mid-Pleistocene forms generally have larger brains, more rounded skulls, and faces that, while still robust by our standards, are trending toward the configuration seen in later humans. Their limb bones suggest efficient bipedalism and bodies that could dissipate heat in open, hot environments. Relatively long limbs, barrel-shaped chests, and reduced body hair inferred from skin and sweat adaptations in our own lineage. Those traits make sense in a world of expanding open habitats, more persistent walking and running, and larger home ranges. A hominin that needs to search widely for waterholes as climates dry, or track mobile herds across patchy grasslands, benefits from a frame that keeps it cool and efficient over distance. At the same time, some fossils in cooler or more temperate regions show hints of stockier builds, thicker bones, and features that foreshadow the even more cold-adapted Neanderthals to come. Enlarged nasal regions that may have helped warm and humidify cold air, strong muscle attachments suggesting powerful upper bodies useful for close-quarter hunting or dealing with heavy tools and carcasses. These physical differences likely reflect local adaptations to different climates and ecologies. They signal that by this point our genus was not a single, uniform type spread thinly over the map, but a set of related populations experimenting with different ways of being hominin in different environments. And that matters when we try to link the fossil world to the genetic signals of a bottleneck. One simple story would be, there was one main ancestral hominin lineage. It went through a massive global crash down to around 1,000 breeding adults, and everyone alive today descends from that tiny group. If that were the whole story, you might expect to see a neat, sharp contraction in both fossils and artifacts around the same time everywhere. Instead, the fossils hint at something more complex. Around and after the supposed bottleneck window, we see evidence of multiple contemporaneous lineages or regional forms. In Africa, populations with Heidelbergensis-like traits. In Europe, possible antecessor-like or early Heidelbergensis forms. In Eastern Asia, enduring Homo erectus populations with their own distinctive skull shapes, and likely other as yet unrecognized or poorly sampled groups in regions we barely have fossils from. Some of these lineages may have been long-term residents, others repeated short-lived expansions. Some may have exchanged genes when climate opened corridors. Others may have remained relatively isolated for long stretches. In a world where barriers and bridges came and went with the ice, species boundaries may not have been as tidy as our textbooks suggest. From a genetic point of view, that mosaic can produce signals that, when viewed only from modern DNA, look like a bottleneck even if the reality was a tangle of splitting, shrinking and merging populations. If one subset of that network later contributed disproportionately to our ancestry, because it survived while others faded, or because its descendants expanded under favourable conditions, their demographic history would dominate the genetic story we can read today. But the bones themselves don't tell us effective population sizes. They don't come with labels saying member of the lineage that will lead to Homo sapiens versus member of a side branch that will leave no descendants. They are snapshots, not a continuous film. This is the limit of what fossils can do for the bottleneck question. They can show us that our genus was widespread, that it was morphologically diverse, and that there were likely multiple overlapping hominin lineages experimenting with different body plans and ecological niches during this turbulent period. They can suggest routes of dispersal, coastal plains when sea levels fell, river corridors through drylands, mountain foothills that held onto moisture, and adaptations that hint at how different populations solve the problems of heat, cold, and scarcity. What they cannot do, at least not yet, is provide a precise head count or cleanly map those bones onto the particular ancestral line that leads directly to us. They can tell us that the hominin world was busy and varied when our genomes say it was constricted. Reconciling those two pictures, outer diversity and inner genetic narrowness, requires turning to another line of evidence. Not just the bodies our ancestors left behind, but the things they made and used with their hands. If fossils are the bones of the story, archaeology is everything our ancestors did with their hands and then accidentally left behind. Across that 1,000,000 to 800,000 year window, the most persistent witnesses are stones, flakes, cores, and battered cobbles that once sat in someone's palm as they struck off an edge sharp enough to cut meat or work wood. At first glance, it doesn't look like much. Simple cores, simple flakes, repeated over hundreds of thousands of years. But if you look closely, those stones whisper things that bones alone can't. Cut marks on animal bones, for instance, are like freeze frames of behaviour. Fine, V-shaped grooves scoring a rib or femur show where a sharp edge sliced through skin and flesh. If those cut marks overlay tooth marks from carnivores, they suggest hominins were scavenging carcasses after big predators had already fed. If they underlie the tooth marks, hominins may have had first access, hunting or at least aggressively stealing kills. At some sites in Africa from roughly this period, you see systematic butchery patterns, repeated cuts at joints to disarticulate limbs, spiral fractures on long bones that indicate the bones were fresh when they were broken open for marrow, piles of flakes and cores sitting right where the carcasses were processed. That points to a strategy: following herds, finding or making waterhole ambushes, transporting at least high-value parts of carcasses back to favoured spots. In other places the pattern looks different. Smaller animals, more diverse prey, fewer heavy butchery marks. Here hominins may have leaned more on broad-spectrum foraging. Small game, plant foods, opportunistic scavenging when the chance arose. The stone tools are still simple, but the way they're used tells you about diet, risk and mobility. Even the tools themselves carry traces of skill and planning. In the earlier Oldowan tradition, a suitable cobble might be flaked in an ad hoc way, producing a few sharp edges and a somewhat battered core. By the time you reach the Acheulean, the hand axe tradition that spreads widely after about 1.7 million years ago and persists well into our bottleneck window, you see something else. Large cutting tools shaped bilaterally, sometimes with a symmetry so precise it borders on aesthetic. Acheulean hand axes show up across much of Africa and Eurasia during this time. They're big, heavy-duty knives that can skin an animal, slice meat, chop wood, dig up tubers. Some sites show standardized forms and careful thinning that imply teaching, practice, maybe even shared mental templates for what a good tool should look like. If you view the period only through genetics, you might picture a population squeezed so tightly that it was clinging on by its fingernails. But the archaeology complicates that picture. In many regions, the stone tool traditions look remarkably stable. For hundreds of thousands of years, hominins kept making more or less the same kinds of Acheulean tools with local variations, but no abrupt universal collapse of technology that would scream demographic catastrophe. In some areas, toolkits even become more refined, thinner, more symmetrical hand axes, more careful selection of raw material, perhaps hints of regionally distinct styles. That stability can be read two ways. One reading is stagnation. Maybe small, isolated populations lack the numbers and social networks to innovate. Another equally plausible reading is robustness. If a toolset works across droughts, cold snaps, and shifting animal ranges, there's no compelling reason to reinvent it. Reliability can be a survival strategy in itself. In a harsh, variable Pleistocene world, a conservative technology might signal that hominin groups had found a versatile generalist solution, a set of tools and behaviours that could be flexed and adapted from river valley to savanna edge to woodland fringe without needing radical overhauls every few thousand years. From that angle, the long Acheulean plateau looks less like a flat line and more like a long, steady hum of competence. But here, as with fossils, we run headlong into the limits of what we can actually see. Archaeological sites don't just sit there waiting for us. They have to survive the same gauntlet as bones: burial, erosion, chemical weathering, tectonic shuffling. On top of that, stone tools are often small, and early hominins were highly mobile. Put those together, and you have a recipe for an archaeological record that is thin, scattered, and profoundly biased. Consider sea levels. Over the last couple of million years, global sea level has risen and fallen by more than 100 meters as ice sheets advanced and retreated. In glacial periods, vast continental shelves were dry land. Coastal plains, estuaries, river mouths. Those are exactly the kinds of places mobile foragers might favour. Rich ecotones full of game, shellfish, plant foods, and fresh water. When sea level rose, those landscapes drowned. Any campsites, butchery zones, or napping spots they held were submerged, buried under layers of marine sediment, or ripped apart by coastal erosion. Today, our best-known early and middle Pleistocene sites cluster inland, or in places where uplift has raised old shorelines. But that doesn't mean hominins weren't using now-submerged coasts heavily. It just means their traces there are largely invisible to us without underwater archaeology on a scale we've barely begun. Erosion cuts the record in the opposite direction. Some landscapes are simply too active, rivers that scour their banks, slopes that collapse, deserts where wind abrades and moves everything. In such dynamic settings, small scatters of stone can be dispersed, buried too deep, or destroyed outright. Other landscapes, like certain lake margins or caves, act as traps. They accumulate sediments, gently burying and preserving tools and bones over long periods. So just as with fossils, our archaeological windows are clustered in a few favourable environments. Caves, ancient lake basins, river terraces that happen to be cut just right. vast amounts of behaviour played out on uplands, floodplains and coastlines that are now eroded away or structurally buried under younger deposits. Sampling bias adds another layer. Some regions, parts of East Africa, bits of Europe, a few basins in China, have seen decades of intensive fieldwork. Teams walk grids, test trenches, map tool scatters. Other regions that may have been crucial corridors or refuges during climate swings, large parts of Central Africa, Arabia, much of South and Southeast Asia, have barely been touched, or their ancient surfaces lie hidden under thick vegetation or modern soils. In a world where our main evidence is stones and bones, geography becomes destiny. Where we can't look, we can't know. All of this matters when we try to picture what a bottleneck world would actually look like on the ground. If the effective population feeding into our ancestry really did shrink to around 1,000 or so breeding individuals at some point in this window, those people were not necessarily clustered in a single camp or valley. They might have been spread thinly across an enormous area, broken into small mobile bands that rarely exceeded a few dozen people each. Groups like that don't leave monumental archaeological signatures. They don't build cities or even large long-lived villages. They may use the same stream bank, rock shelter, or knoll seasonally, returning year after year, but with light footprints each time, a few hearths, a modest scatter of flakes, some broken bones, maybe a handful of hand axes cached for later. On an open plain, wind and water can erase such traces quickly. On a river terrace, they may be buried and later re-exposed, jumbled together with traces from different visits and different millennia. In a cave, they might accumulate into thicker deposits we can actually excavate. But caves are rare. Most of the landscape is not a cave. Imagine trying to reconstruct the human population of the modern world if you were allowed to see only what was left in a few dozen caves and gullies. You might conclude that humans were always clustered in those spots, that certain tool styles dominated simply because that's what those particular groups used, that empty spaces on your map meant empty land rather than absent evidence. Now add one more twist. Small mobile groups can be demographically fragile and culturally resilient at the same time. A band can hover near the edge of extinction, one bad drought, one epidemic, one run of poor hunting away from vanishing, and still maintain a remarkably consistent way of making tools and processing food across generations. Knowledge can be passed down even in tiny communities, especially if the core tasks and materials remain the same. That's the core tension we keep running into. Modern DNA, modelled through various demographic scenarios, seems to point toward a long period when the ancestral population that would eventually lead to us was unusually restricted. Like a river forced through a narrow gorge. It doesn't necessarily mean other hominin lineages elsewhere weren't doing their own thing. It means that from the vantage point of our genomes today, one thread in that web carried forward most of the ancestry. The material record from 1.2 million to 800,000 years ago, by contrast, looks more like a wide, braided channel, overlapping distributions of tools and sites across Africa and Eurasia, persistent Acheulean traditions, local tweaks rather than dramatic breaks, cut-marked bones that point to varied but functioning survival strategies. Is that because the genetic bottleneck was subtle on the ground, playing out as slight shifts in group sizes and connections that archaeology is too coarse to resolve? Or is our reading of the genetic signal shaped by assumptions about population structure and migration, oversimplifying a more complex reality of many semi-isolated populations whose fates rose and fell asynchronously? Right now, archaeology can't answer that definitively. But it does remind us of two important things. First, absence of evidence is not evidence of absence. A sparse scatter of sites does not automatically mean a sparse scatter of hominins. Tiny, mobile populations on eroding, shifting landscapes are almost designed to vanish from the record. Second, cultural continuity is not the same as demographic stability. A seemingly unchanging toolkit can coexist with dramatic swings in population size and connectivity. The Acheulean hand axe may have been a remarkably adaptable technology that weathered booms, busts and bottlenecks without needing to transform. To untangle climate, catastrophe and population structure in this bottleneck story, we have to carry both pictures in our heads at once, genomes that remember a squeeze and stones that tell a more equivocal, patchy tale of persistence. The next step is to ask whether the physical world itself, ice sheets, rainfall belts, oceans, could have forced our ancestors into that genetic narrow passage. If you want to imagine a force big enough to squeeze a whole lineage, climate is an easy place to start. Between about 1.2 million and 800,000 years ago, the planet was not calmly humming along in the background. The Ice Age rhythm that had been ticking in 100,000-year cycles began to shift toward longer, deeper glaciations. Ice sheets in the higher latitudes grew more massive, sea levels lurched up and down by tens of meters, and the great atmospheric heat engine that drives monsoons and storm tracks became jumpier. In a climate-driven bottleneck scenario, you don't need one single apocalyptic event. You just need pulses. A bad decade here, a harsher century there, repeated often enough that the old reliable patterns of rain and temperature fragment into something more like a strobe light. For hominins whose survival hinged on water and seasonally dependable food, those pulses would have translated directly into the size and shape of their world. Paleoclimate records from ocean sediments and ancient lake beds tell a recurring story. Wet phases when lakes in East Africa swelled, grasslands flourished and woodlands thickened, followed by dry intervals when those lakes shrank or vanished, rivers turned intermittent, and open, dusty plains spread. In some reconstructions, the amplitude of those swings increases through exactly the window when the genetic bottleneck is inferred. Imagine living inside that curve. In a relatively stable period, hominin groups might map their movements around a web of known water sources, perennial springs, reliable river bends, long-lived lakes. Around those hubs, animal herds rotate, plant foods track the seasons, and the route from dry season refuge to wet season grazing follows a pattern your grandparents and their grandparents already knew. Now imagine that over a few generations that web phrase, a lake that always held water through the dry months fails, not just once but several times in a row. A river that used to flow year-round becomes seasonal, its dry season pools smaller, crowded with desperate animals. The strips of woodland that buffered you against drought retreat upslope or disappear. The safe path between two valleys, once dotted with predictable stopovers, becomes a gauntlet of longer, riskier marches. This is what ecologists call habitat fragmentation in slow motion. Not walls and fences overnight, but the gradual loss of continuous usable space. For a thirsty terrestrial primate with limited ability to store water, that matters. Water is the first pinch point. Hominins can stretch food scarcity. They can fast, fall back on low-quality staples, or move farther to find game. But without water, range collapses quickly. If perennial sources shrink into scattered refugia, deep lakes, certain springs, river reaches fed by unusually resilient catchments, then any population that wants to survive the worst dry seasons is effectively funneled into those shrinking oases. The second pinch point is timing. Even in lush ecosystems, food is not evenly available year-round. Animals carve and fatten on their own cycles. Fruits ripen in particular months. Tubers and underground storage organs may be more accessible in one season than another. Hominins of this period had already learned to juggle such seasonal bottlenecks, but repeated climate swings can distort the calendar. Rains may arrive late or fail 2 or 3 years in a row. The plant species your group leans on for lean season calories might wither or shift their range upslope faster than you can track them with a forager's memory. A migration you once counted on, say a herd that always passed within reach of a particular valley, might now bypass your territory entirely, tracking greener pastures along a different corridor. String enough bad seasons together, and the margin between hungry but coping and too weak to move when you need to thins. Under those conditions, malnutrition doesn't just mean weight loss, it means compounding risk. Chronic underfeeding weakens immune systems, injuries heal more slowly. Pregnancies are riskier, fetuses may be smaller, more vulnerable. Infants weaned onto poor quality foods are more likely to die of infections that a better fed body might shrug off. Small children, who need dense nutrition to support rapidly growing brains and bodies, are especially exposed. Disease in that sense is not a separate catastrophe but a multiplier. Crowding at the few remaining water sources brings more individuals and more species into close contact. Pathogens circulate in those stressed communities more easily. Parasites and waterborne illnesses thrive where animals, including humans, are squeezed around dwindling pools. We don't have direct evidence of specific epidemics in this period the way we do for historic plagues. But the logic is simple. Stressed bodies in compressed spaces are fertile ground for microbes. Now transpose that onto the demographic canvas suggested by genetics. If the ancestral population feeding into later humans was already not enormous, scattered into many small bands spread across Africa and perhaps nearby regions, then repeated climate pulses could prune that network in a patchwork way. A valley that loses its last reliable spring might see its bands abandon it, perish, or merge with neighbours. A corridor that once linked two regions becomes an arid barrier during the toughest glacials, cutting gene flow. A highland refuge that stays just wet enough could preserve a cluster of groups that, by sheer luck of geography, end up overrepresented in our later ancestry. Climate models and paleoproxies suggest such refugia probably existed. Pockets where local topography or ocean atmosphere quirks buffered the worst extremes. Parts of East Africa, some river systems in Central Africa, certain coastal or montane zones may have acted as demographic lifeboats. In a climate-driven bottleneck story, those lifeboats are where the 1,280 effective individuals would be hiding in plain sight, walking, hunting, raising children, and passing on their genes while vast areas between refuges turn marginal or temporarily uninhabitable for hominins like them. This is a powerful picture. It doesn't depend on a single bolt of lightning from the sky. It leans on a process we know was absolutely in play: Ice Age climate variability reshaping habitats, and water in ways that matter tremendously to large mammals. But as with everything in this story, it's when you zoom out that trouble appears. One issue is scale. Even under quite extreme climate scenarios, Africa and adjacent regions do not turn into a single, uniform desert or glacier. Wet belts shift. Some rainfall corridors move north or south. Highlands can catch moisture that plains miss. Deep rift lakes may hold water when shallower basins blink in and out. From the perspective of a single small band, climate-driven hardship might feel like the end of the world. From the perspective of an entire continent, it looks more like a chessboard of good squares, bad squares, and changing pathways between them. And hominins of this period were not confined to one tiny corner of that board. Fossils and tools show representatives of our broader lineage in parts of East and South Africa, North Africa, the Levant, perhaps Arabia and beyond. Some of these regions would have been in drought while others had comparatively mild conditions. Some would have lost key habitats. Others gained them as shorelines moved and vegetation belts shifted. That geographic spread acts as a buffer. Climate can push populations around, shrink them in one area, and expand them in another. but driving a deep, long-lasting bottleneck across everything at once is a harder trick. Another issue is resilience. Large mammals, especially generalists, often respond to climate stress not just by dying, but by moving. Hominins were mobile, flexible omnivores. They could tweak diets toward tougher plant foods, pursue different prey, alter group ranges, or explore new corridors along rivers and coasts. If the climate story alone is responsible for the genetic bottleneck, we have to imagine that those adaptive levers were not enough. That, despite the behavioral flexibility, the repeated pulses were severe and synchronized enough across hominin ranges to keep effective population size low for hundreds of thousands of years. That's not impossible. Some modelling studies argue that long periods of heightened climate volatility can cap population sizes even without killing everyone in sight, simply by keeping carrying capacity low and variable. Under that view, you don't need one giant crash. You need a long era when populations never really get to boom. But we also have to account for the fact that other large mammals, including some quite climate-sensitive species, seem to have threaded these same swings without showing such a dramatic, prolonged genetic squeeze, and that other hominin lineages, those that would later contribute to Neanderthals, Denisovans, and perhaps still unknown groups, were playing out their own histories across Eurasia. If climate alone was the executioner, why does the most obvious bottleneck signal appear so strongly in the ancestry leading to us, and not as clearly and simultaneously everywhere else? One possible answer is that our particular branch just happened to be tethered to habitats and corridors that were especially vulnerable, say certain East African lake basin mosaics that were hammered by these swings. Another is that what genetics is seeing is not only raw numbers, but changes in how subpopulations were connected. Climate might have fragmented groups into more isolated pockets, making them behave, from a genetic standpoint, like a smaller population even if the headcount was higher. Yet even with those nuances, many researchers suspect that climate, while major, may not have acted alone. A drying or cooling world can set the stage, pushing groups into refugia, fragmenting habitats, upping the stakes around waterholes and migration routes. On that stage, other actors, competition with rival hominins, shifts in predator guilds, outbreaks of disease, even geological surprises, can amplify or dampen the bottleneck effect. There's also a subtler demographic angle. Climate stress can change social behaviour. Bands might fuse or fission differently, adjust mating patterns, alter how far young adults disperse. Those shifts can have surprisingly large effects on effective population size, the genetic shadow of a population, without necessarily altering the raw census as dramatically. So could climate alone have forced our ancestors through that narrow genetic gorge? It's certainly one of the most compelling candidates. The timing lines up broadly. The mechanisms, shrinking refugia, water and food bottlenecks, chronic stress and disease, fragmentation of ranges, are plausible and consistent with what we know about ecology in Pleistocene environments. But the same evidence that makes climate attractive also makes it hard to isolate. Climate touches everything. Any other process you propose, competition, isolation, catastrophe, is almost certainly mediated by, or entangled with, shifting rainfall and temperature. In the end, most scientists who study this problem tend to treat climate not as the lone culprit, but as the backdrop and the pressure cooker. It can explain why life got harder, why refugia mattered, why some lineages may have withered while others endured. Whether it's sufficient on its own to explain a bottleneck as deep and prolonged as the one some genetic models infer remains an open question. To press further, we have to look at what else was happening in that restless world. The eruptions, the tectonic reshaping, the other hominins on the landscape, and the quieter dynamics of small scattered groups drifting in and out of contact as the Ice Age pendulum swung. If climate is the long grinding pressure, Volcanoes are the tempting villains, the sudden plot twist that change everything overnight. Not all eruptions are equal. Many volcanoes spend their lives coughing up modest amounts of lava and ash that matter a great deal if you live on their slopes, but barely register on a continental scale. For our ancestors, those kinds of events could still be lethal, burying local valleys, poisoning streams, smothering grazing lands. A big regional eruption can erase habitat, wipe out herds, and force wandering bands into already crowded refuges. But there's another class of eruption that grabs the imagination, the truly large explosive events that punch ash and sulfur high into the stratosphere. These don't just coat the neighborhood, they alter how much sunlight reaches the surface. When enough sulfur dioxide gets lofted high and spread wide, it forms reflective aerosols that dim the sun, cool the ground, and shuffle rainfall patterns. The result is what's sometimes called a volcanic winter, shorter growing seasons, crop failures in agricultural societies, stressed ecosystems even in the deep past. We have a recent example humanity has actually lived through, in historical terms, the Toba supereruption in Indonesia about 74,000 years ago. It ejected thousands of cubic kilometers of material, an almost incomprehensible volume, and left a caldera tens of kilometers across. Climate proxies suggest it did send a pulse of cooling through parts of the world, and ash from that event appears in layers from the Indian Ocean to Africa. For a long time, some researchers argued that Toba might have nearly exterminated Homo sapiens, leaving only a tiny remnant population, a bottleneck that would show up in our genes. It was an appealingly simple story. Giant eruption, global volcanic winter, human numbers crash, survivors repopulate. But as more archaeological and genetic evidence accumulated, that neat picture frayed. Sites in Africa and South Asia show continuity in stone tool traditions across the Toba layer. People were there before and after, apparently adapting rather than collapsing. Genetic studies also failed to find a dramatic, tightly dated bottleneck at 74,000 years ago in our species. Any demographic dips related to Toba, if they occurred, were more modest and regionally uneven than the early catastrophe model suggested. That Toba story is a cautionary tale, and it matters for our much older puzzle. The inferred bottleneck that might have squeezed our lineage down to an effective size of about 1,280 individuals doesn't sit around 74,000 years ago. It sits way back, in the broad band from roughly 900,000 to 800,000 years ago. So if a volcano, or any single disaster, is going to be blamed for that event, it has to live in that window and leave fingerprints that match the scale and duration of the genetic signal. There were certainly eruptions then. The East African rift, where many hominins lived, is volcanically active. Italy's volcanic provinces, the Andean and Indonesian arcs, and scattered other centres would have been rumbling as well. Over hundreds of thousands of years, it would be more surprising if there weren't some very large eruptions. The difficulty is connecting any one of them cleanly to the genetic story. Part of the problem is basic dating. Genetic inferences about population size through time are inherently fuzzy. They don't spit out a crash occurred exactly 897,413 years ago. They produce broad intervals with uncertainty ranges spanning tens or even hundreds of thousands of years. Volcanic deposits can be dated too, with radiometric clocks in minerals, with paleomagnetic markers, with stratigraphic relationships. But those methods also come with error bars. To argue that a particular eruption triggered our bottleneck, you'd need those independent clocks to line up convincingly. So far, they don't. There is no widely accepted, globally obvious supereruption that is both securely dated and perfectly aligned with the onset of the putative 900 to 800,000 year bottleneck. Even if there were, another snag appears: scale and shape. A truly massive eruption is, by definition, a sudden shock. The climate and ecosystem disruption might last years, perhaps a decade or two in extreme scenarios, with some downstream effects that echo for centuries. Trees die, grasslands shift, animal populations slump, and then, if conditions allow, ecosystems rebuild. For agriculture, a few years of failed harvests are catastrophic. For small foraging populations spread across continents, a bad decade is deadly but not necessarily permanently population limiting. The genetic bottleneck, as some models reconstruct it, doesn't look like a short, sharp dip that rebounds quickly. It looks more like a long, low ceiling on population size, maintained for hundreds of thousands of years. That's closer to a drawn-out period of ecological constraint than to a single hammer blow. Volcanoes are good hammers. They are less convincing as tools for keeping populations depressed for 500,000 years. There's also the question of geography. Eruptions, even very big ones, are fundamentally regional. Ash clouds spread in preferred directions dictated by wind patterns. The most severe fallout, lahars and lava, are limited to areas downwind or downslope. A hominin population living on the flanks of an East African volcano would experience a catastrophe very differently from one living in West Africa, or along a distant coastline, or in the Levant. If a regional disaster is to be blamed, its predictions are very specific. You'd expect local or regional population crashes where ashfall was thick, water sources contaminated, and prey bases devastated. Refugia in areas outside the main impact zone, where hominins could persist with less disruption. A genetic signature showing that later populations trace disproportionately back to those refugial regions rather than to the obliterated zones. That kind of patchwork survival is actually compatible with how genetics reads a bottleneck, not as a uniform thinning everywhere, but as the overrepresentation of a subset of lineages that happened, by chance or geography, to survive. Theoretically, a catastrophic eruption that wiped out most groups in a core hominin range while sparing some outlying pockets could contribute to a sharp reduction in effective population size. But for this to explain a deep sustained bottleneck, it can't just be one bad day. Either the same region would have to be hit repeatedly by large eruptions as soon as it started to recover, or those eruptions would need to interact with other stresses, climate volatility, competition, habitat fragmentation, in ways that kept carrying capacity low for a very long time. In other words, volcanoes start to look less like solitary culprits and more like accomplices in a large conspiracy. The contrast with a truly global catastrophe is instructive. Consider a hypothetical impact event big enough to inject dust and aerosols worldwide, darkening skies for years and tanking plant productivity across continents. That kind of disaster, unlike a regional eruption, would hit hominin populations simultaneously in many different regions. Its genetic fingerprint might be a more synchronized sharp bottleneck across multiple lineages, not just in the ancestors of modern humans, but in Neanderthal Denisovan ancestors, in other African hominins, maybe even in large mammals more broadly. We do not see such a universal, neatly aligned crash in the available genetic records. Other lineages have their own ups and downs, but not a single, obvious, global wipeout in that 900 to 800,000 year window. Large mammals did not vanish en masse. That doesn't rule out significant disasters. It does argue against a clean, earth-spanning apocalypse tailored just to our branch. There's another subtler reason to be cautious about pinning the bottleneck on a specific catastrophe: the shape of genetic memory itself. Genetic bottlenecks record who left descendants, not every death. A disease outbreak, a volcanic winter, or a mega-drought can slash numbers. But if many different lineages survive and rebound, the long-term effective size may not plummet as dramatically as the census did in the crisis moment. Conversely, you can get a strong bottleneck signal without a single spectacular event, simply by having a long era during which only a limited set of interconnected groups contribute most of the genes that eventually lead to us. Disasters, in that sense, are easiest to spot when they are numerous, biased, and patterned, when they repeatedly prune the same parts of the hominin family tree and spare the same refugia. A volcano that happens to explode in a demographic backwater leaves little genetic echo. A volcano that hammers the central hub of a species range in a time when climate and competition already have that species on the ropes might leave a deeper mark. At present, we don't have the granular resolution to trace such chains of cause and effect. We can point to periods when volcanic activity, tectonic uplift, and climatic shifts all overlapped. We can imagine scenarios in which eruptions briefly shut down key corridors, dirty vital lakes, or tip already stressed ecosystems across a threshold. What we cannot do yet is say, this caldera here, this ashbed dated to this year, is the spark that set off the 1,280 individual bottleneck. Most researchers, therefore, treat large eruptions and kindred disasters as plausible contributors rather than proven drivers. They may have sharpened the edges of climate stress, turned marginal years into lethal ones, and reshaped regional population maps. They give us vivid stories of danger and survival, but in the careful language of science they sit in the maybe and probably as part of a bigger picture column. That bigger picture includes forces that don't make headlines as easily as exploding mountains. It includes the ways small scattered groups can become genetically isolated without any obvious catastrophe at all, simply by drifting apart across fragmented landscapes, losing contact, and then, over long stretches of time, leaving only a few threads that lead forward to us. To see how that might work, shift the camera in your mind from erupting volcanoes and collapsing ice sheets to something much less dramatic: distance. Not a heroic trek across continents, just the ordinary, accumulating distance between one valley and the next. A ridge that takes a day to cross instead of an afternoon. A river that floods deeper and longer in the wet season. A stretch of scrub that offers little shade and less water, so people don't cross it unless they really have to. Now imagine small bands of hominins threaded through this kind of patchy landscape. A few dozen individuals here, a few dozen there. They may all speak similar calls, crack similar stones into cutting edges, and hunt the same kinds of animals, but they don't see each other very often. Weeks can pass, seasons, years, without one group meeting another. Babies are born, grow up, and have children of their own without ever ranging far beyond the edges of their home territory. In that kind of world, you don't need a catastrophe to shrink the effective population size. Geography and behaviour can do it for you. When geneticists talk about effective size, they're not counting how many bodies are walking around. They're asking a different question. How many of those bodies are actually mixing their genes with one another through mating, across the whole species, over time? In a single, large, well-connected population, that number and the head count are roughly similar. But as soon as you break that population into pockets that rarely exchange mates, the effective size drops, sometimes dramatically, even if the census stays high. Think of it like this. A species spread across a continent might actually be living as a mosaic of semi-isolated neighbourhoods. If each neighbourhood mostly marries within itself, then from the gene's perspective, it's as if there are several small micro-populations instead of one big one. In such a scenario, random events hit harder. A bad hunting year, a disease, a predator boom. All of these can wipe out or shrink one pocket without touching the others. Over time, some neighbourhoods blink out completely, while others keep going. Now, zoom in on the climatic chaos of 1.2 million to 800,000 years ago. Glacial cycles are intensifying. Rain belts wobble. Uplift and erosion are reshaping river basins and highlands. Grasslands expand here and retreat there. Forests fragment into groves on favoured hillsides and along certain rivers. Instead of a broad, continuous belt of good habitat, you get a broken chain of what ecologists call refugia, scattered safe zones, where conditions remain just tolerable enough for a species to hang on. In that refugia model, our ancestors aren't spread smoothly across Africa and Eurasia. They're clumped into islands of relative comfort, a Rift Valley lake with stable water levels, a coastal strip where upwelling seas still feed rich shellfish beds, a plateau that catches just enough rain to grow edible plants and draw in grazing herds. Between those pockets lie harsher stretches, too dry, too cold, too unpredictable to support permanent communities. As climates swing, the locations of those refugia can shift. A once reliable lake may shrink into a briny puddle. A river might change course, abandoning one floodplain and blessing another. In response, hominin groups move if they can, track the resources, squeeze into the next livable patch. But they don't all move together in lockstep. Some are cut off. Some never hear about a better valley over the ridge. Some try to cross the widening deserts or the thickening forests and fail. From a genetic standpoint, this is like repeatedly shaking a box of marbles and watching some spill out each time. Each refugium holds only a fraction of the original diversity. When a local group is founded by just a few individuals who manage to make it to the new safe zone, their descendants carry only a subset of the variants from the larger ancestral pool. That's the founder effect. A thinning of genetic variation, not because the species nearly died out everywhere, but because only a small sample of its diversity got to seed each new pocket. Stack enough founder events in sequence, groups splitting off, moving into new refugia, surviving while others fail, and the effective population size falls. It's no longer the sum of everyone alive. It's the sum of the lineages that keep successfully hopping from one safe patch to the next over long periods of instability. Now add another layer, extinction that is local, not global. In a fragmented world, some refugia will be dead ends. A band hanging on in a marginal valley might do fine for a few thousand years, then hit a streak of bad luck. A drought that lasts just a little too long, a disease that sweeps through at the wrong moment, a competitor that moves in and dominates the best water holes. That group's line simply stops. Their genes vanish from the future, even though other hominin pockets safely tucked elsewhere keep going. If this pattern repeats, some pockets persist, others wink out, you get a kind of genetic sieve. Over hundreds of thousands of years, many branches bud and die without leaving a mark in modern genomes. Only a limited subset of regions and groups keep contributing to the long-term ancestry that leads eventually to us. To a geneticist looking backward, it can resemble a narrow bottleneck, a small effective population feeding the future, even if the total number of living hominins at any given time was substantially larger. This is where isolation and later reconnection create an illusion of dramatic booms and busts in genetic reconstructions. Imagine 2 or 3 major refugial regions, each with their own modest, semi-stable hominin populations, say tens of thousands scattered across Africa and perhaps adjacent areas, but broken into many isolated bands. For a long stretch, those regions barely exchange genes. From the perspective of methods that reconstruct past population sizes, that looks like a single, relatively small, effective population, because only within refuge mixing is common. Then, conditions change. Climate becomes somewhat more forgiving. A key corridor opens, perhaps a wetter stretch through what was once a barrier desert, or a chain of lakes that link 2 basins. Groups that have been evolving largely apart for hundreds of thousands of years now start to meet again, tentatively at first, then more often. Mates are exchanged, children are born who carry mixed ancestries from formerly separate pockets. Gene flow resumes across what had been a patchwork of near isolates. To the genetic reconstructions, this reconnection looks like a rebound. The effective population size seems to increase, not necessarily because the head count suddenly skyrocketed, but because the neighbourhood in which genes can meet and mingle has expanded. Variants that were once trapped in regional silos are now part of a larger, more cosmopolitan pool. Diversity rises, coalescent models infer that more lineages were contributing, and the graph of population size through time shows a climb. Crucially, nothing in this story requires the species as a whole to have been teetering on the verge of extinction, with a single tiny band of 1,280 desperate survivors huddled in one cave. You can get a similar long, low plateau in effective size through a quieter combination of fragmentation, limited gene flow, localized founder effects, and repeated small-scale extinctions that prune the tree unevenly. Some researchers argue that this kind of structure is not just a detail, but may be central to interpreting the deep bottleneck signal. If our ancestors were living in a chronically patchy world, corridors opening and closing, bands fissioning and fusing, refugia appearing and disappearing, then the genetic record might be telling us more about the connectivity of those networks than about a single catastrophic downturn in absolute numbers. That doesn't mean dramatic crashes never happened. It means that, superimposed on any crashes, there may have been a persistent background of isolation, a species spread thinly across difficult terrain, rarely unified into one big breeding population. In such a setting, population structure itself behaves like a quiet sculptor, carving down effective size even as local census numbers rise and fall. As an alternative to the one big disaster narrative, this view has an appeal. It fits with what we know of other animals living in highly variable environments. They often survive not as continuous blankets across the landscape, but as shifting clusters in favoured pockets. It also dovetails with the increasing recognition in human origins research that our ancestors were probably never a single homogeneous block, but a braided stream of semi-separate groups, sometimes entwining, sometimes diverging. Still, structure and isolation alone may not be the whole story. Fragmentation raises questions of its own. What forces kept groups apart? What pressures repeatedly trimmed their numbers? And how did those pressures interact with competition from other hominins, predators, and changing food webs? To understand that, we have to zoom out again, not to volcanoes this time, but to the entire ecological theatre in which these small, scattered bands were trying to make a living. When you zoom out to that ecological theatre, the first thing you notice is that our ancestors were not the main characters. On most landscapes between 1.2 million and 800,000 years ago, big mammals and big carnivores are still running the show. Hominins are present, sometimes widespread, but they're more like persistent supporting actors trying to carve out a stable role in a cast that keeps changing. And that cast was changing in ways that could quietly squeeze hominin lives. Across Africa and Eurasia, this was an age of turnover in large mammals. Species of elephants, hippos, buffalo, antelope, and even big cats appear, flourish, and vanish on timescales of hundreds of thousands of years. Some are replaced by close relatives with slightly different habits. Others leave ecological gaps that are filled in new ways. Climate is one driver. Shifting rainfall and vegetation patterns favour different grazers and browsers, but the consequences ripple far beyond plants and herbivores. For a hominin band trying to make a living off meat, fat and marrow, what matters is not just are there animals, but which animals, how many, how dangerous and how predictable. Imagine one valley where huge, slow-moving, water-dependent herbivores dominate the scene, perhaps ancient relatives of hippos and elephants, tied to river margins and lake shores. Their movements are constrained, their carcasses massive rewards when found or brought down. Now imagine that, over a few tens of thousands of years, drying trends favour more mobile grazers, smaller antelope, wild cattle, zebras, animals that range widely across patchy grasslands, appearing and disappearing with the rains. For lions or hyenas, that shift might simply mean adjusting hunting tactics. For hominins with simple stone tools and no projectile weapons, it can fundamentally alter the economics of survival. Big, predictable carcasses near water become rarer. Smaller, faster prey dominate, and many of those species are harder to approach and more dangerous to confront up close. The odds of finding a half-eaten elephant left by lions drop. The odds of running into these lions far from shelter stay high. This is where competition really bites. Hominins at this time were almost certainly not top predators in the way we often imagine hunters. They were opportunists, sometimes hunting small and medium game, often scavenging from kills made by carnivores. In that niche, they were stepping into a crowded arena. Think of the carnivore guild, saber-toothed cats fading but not gone at the beginning of this interval. Big pantherine cats like early lions and leopards, spotted and striped hyenas, wild dogs, perhaps even large eagles and other birds of prey. All of them evolved teeth, claws, speed, and coordination precisely tuned to tracking, killing, and defending meat on the landscape. Hominins brought a different toolkit. Hands, cooperation, simple stone blades, and heavy pounding tools. A brain that could remember where carcasses tended to appear and how to read the behaviour of scavengers. That combination might let them punch above their weight, arriving early at kills, harassing predators off fresh meat with thrown stones and loud bluffing, cracking tough bones that even hyenas struggled with. But the balance between bold opportunist and reckless intruder depends on the exact mix of predators around and on how much meat there is to go around. If environmental shifts reduce the number of large herbivores, then every carcass rises in value for everyone. Lions, hyenas, and wild dogs become less tolerant of competitors. Fights at carcasses get more dangerous. The window of time between a kill being made and being stripped clean of edible tissue narrows. For a hominin band, the same risky behaviour, walking out onto an open floodplain to drive off hyenas, pays off less often and costs more lives. Now add another layer, competition not just with carnivores, but with other hominins. By this period, there may have been multiple hominin lineages overlapping in time and space. Perhaps earlier African Homo populations, early forms often grouped under names like Homo heidelbergensis, and, outside Africa, various descendant lineages that would later give rise to Neanderthals and Denisovans. The taxonomy is debated and the fossil record is thin, but it's unlikely that a single, uniform kind of proto-human had the entire Old World to itself. Different hominin groups might have favoured slightly different strategies. One lineage could be more tied to wetlands and fish, another more focused on large herbivores. One might have experimented a bit more with persistent hunting, or with using fire in limited ways. Even modest differences in behaviour or anatomy, leg length, shoulder flexibility, gut capacity, tolerance for cold, can lead to different ecological sweet spots. Where their territories overlapped, these groups would have been drawing from the same broad resource base. Waterholes, nut-bearing trees, migratory herds, carcass sites, caves, and rock shelters. Unlike lions and hyenas, they may not have been engaged in straightforward lethal conflict very often, but competition doesn't require constant fighting. It can play out as one group consistently reaching key spots first, monopolizing the best sleeping cliffs, or dominating the timing at which safe access to carcasses is possible. If your band tends to arrive second at every good carcass because another hominin group is slightly better at tracking vultures, that's a chronic disadvantage. You don't necessarily die in a single famine year. Instead, your infants are just a little more likely to be underweight. Your adults recover more slowly from illness. Your elders die a bit younger. Over generations, that kind of edge can shift which lineages expand and which contract. Tie this back to changing food webs, and you get a picture of pressure that builds quietly. As climates wobble, the composition of animal communities changes. Predator populations respond, sometimes booming on the back of new prey, sometimes shrinking in lean periods, but always sensitive to the availability of meat and water. Hominins are slotted into this system at a vulnerable point. They are large mammals with relatively slow reproduction, high energy needs, and, at this stage, limited technology. If rainfall patterns become erratic, plant productivity can drop in some years and surge in others. Herbivore numbers may lag behind those shifts, leading to stretches where there are simply fewer big animals to hunt or scavenge. In those intervals, carnivores may turn more often to stealing from one another or patrolling a wider area, running into hominins more frequently. In a fragmented, refugia-based world, some pockets might navigate these changes relatively well, perhaps because their local herbivore community happens to be resilient, or because a nearby wetland buffers the worst of the droughts. Other pockets might be hammered repeatedly by unlucky combinations, a decline in key prey species, a temporary surge in a particularly dangerous predator, and intensified overlap with a competing hominin group following the same shrinking water sources. Notice that none of this requires a sudden global calamity. It's not a volcano covering half a continent in ash, or an ice sheet bulldozing entire ecosystems in a decade. It's a slow ratchet. Each fluctuation in climate subtly rearranges the food web, and each rearrangement forces hominins to adjust where they forage, when they move, how they negotiate with predators, and what risks they accept at carcasses. Sometimes those adjustments work, sometimes they don't. If you live in one of the unlucky refugia, your group might endure several generations of marginal returns on hunting and scavenging. Children survive at lower rates. Adolescents take bigger chances to find food. Young adults die more often in confrontations with carnivores at kills. Your local census might never crash in a single dramatic decade, but across a few thousand years, the population can thin to the point where a bad run of years pushes it below the threshold of long-term viability. From a genetic perspective, that's another local lineage blinking out, another marble spilling from the box. Other refugia with slightly different predator-prey balances remain viable and keep feeding ancestry into the future. To a modern genetic reconstruction, what's visible is the cumulative effect. Fewer long-lived lineages bridging that 1.2 million to 800,000 year window as pockets exposed to unfavorable food web dynamics quietly fail. So how do we know any of this happened? We don't in the strict courtroom sense of proof. What we have are suggestive pieces, fossil assemblages showing shifts in herbivore species through time, evidence that some large carnivores declined or disappeared in certain regions, cut-marked bones indicating that hominins were indeed in the business of scavenging and butchering large animals, tools and site distributions hinting at where hominins focused their activities. We can see that the caste of mammals was changing, that hominins and carnivores were interacting around carcasses, and that ecological roles were in flux. Ecologists can model how changes in prey abundance might propagate through carnivore populations and into the scavenger niche where hominins were operating. From those models, scenarios emerge in which hominin bands face persistent stress without any singular disaster event, but direct links, the kind that let you point to a bone and say, this lineage here declined because lions outcompeted them for zebra carcasses after species X of antelope went locally extinct, are beyond our reach. The time resolutions are too coarse, the sites too scattered, the behaviours too ephemeral. we're inferring a tangled web of interactions from snapshots. That's why these ideas sit in a middle ground, plausible mechanisms that fit with broader ecological principles, yet supported by only fragmentary indirect evidence. They're not wild speculation. They're grounded in what we know about how food webs work today and in the broad outlines of the fossil record, but they're also not smoking guns. At best, competition, predators, and changing food webs offer a way to transform environmental variability into real demographic consequences for hominins, a way to translate "the climate got weird" into fewer babies survived, more adults died, and some lineages quietly disappeared. Layer that onto a world already fragmented into refugia, and you get a recipe for a long, low genetic bottleneck driven not by a single blow, but by thousands of small cuts. If that's even roughly right, it raises an uncomfortable question. What does living under that kind of chronic invisible pressure do to a species over time? Especially to a species whose fate would eventually be tied to big brains, cooperation, and culture. To explore that, we have to turn from the outside pressures to the inside responses and ask what the bottleneck might have changed in us. When geneticists talk about a bottleneck, they're not just talking about a small head count, they're talking about what happens to variation when almost all of your marbles are suddenly gone. Imagine, again, that big box of coloured marbles representing all the genetic variants roaming around in early Homo. Before the bottleneck, the box is crowded, many slightly different versions of genes for immune responses, metabolism, brain wiring, skin and hair, growth rates. Some variants are common, some rare. Some are helpful in certain environments, some are neutral, some are mildly harmful, but tolerated because there are so many bodies, so many births, that evolution can quietly prune them away over time. Now squeeze that population down to something like 1,000 breeding individuals for tens of thousands of years. Immediately, the arithmetic of chance changes. In a large population, random fluctuations in which variants get passed on tend to average out. If 40% of the population carries a particular neutral mutation, births and deaths shuffle that percentage up and down, but not by much. In a tiny population, that same mutation can vanish just because its few carriers happen to die without leaving children, or, equally, it can shoot up to near universality because those carriers by luck have more surviving offspring. This is genetic drift. Evolution by random sampling, not by the steady hand of natural selection preferring the fittest. Drift is always happening, but in a bottleneck it stops being background noise and starts being a main character. That matters for what comes out the other side. Run that long, low bottleneck, and you don't just lose marbles at random. You also lock in a peculiar, thinned-out subset of what was once possible. Diversity drops, especially at parts of the genome that selection isn't actively protecting. Rare variants, some potentially useful in unusual circumstances, are especially likely to blink out. Long continuous stretches of DNA become more uniform because everyone alive descends from the same small pool of ancestors who survived that drawn-out squeeze. The result isn't simply less variation. It's a different landscape of variation. Different combinations of genes that happened by luck to survive. Could that reshaping have anything to do with later, distinctively human traits? This is where it gets tempting to draw straight lines, and where we have to be careful. Some researchers have wondered whether extended periods of low effective population size could have set the stage for things we associate with the later Homo. Changes in brain growth trajectories, shifts in how our chromosomes are organized, alterations in how genes are turned on and off in the developing cortex, or in our immune system's strange tendency to be both powerful and easily misdirected. They don't mean the bottleneck created language or art or symbolic culture. That comes far later, in lineages that have already passed through many other filters. But a long bottleneck could, in principle, have pruned the ancestral gene pool in ways that nudged which raw materials were available when those later innovations evolved. Here's one way to think about it. Take something like brain development. The wiring of a hominin brain isn't controlled by a single intelligence gene, but by cascades of interacting genes that govern when neural stem cells divide, how long different regions keep adding new neurons, how axons find their targets, how synapses stabilize or disappear. In a large genetically diverse population, there might be dozens of slightly different combinations of these regulatory genes circulating, some pushing development subtly toward more prolonged growth phases, some toward shorter ones, some shifting how different areas of the brain scale relative to each other. Most of those combinations are probably good enough. As long as you grow a functional brain that can navigate social life and find food, fine-tuning may not have a huge visible effect on survival in the short term. That makes many of these variants effectively neutral, or only very weakly selected. In a bottleneck, such weakly selected variants start behaving more like passengers than drivers. Drift can fix one developmental setting simply because the individuals carrying it happen to be the ones who made it through the lean millennia. Others are lost, not because they were worse, but because their carriers were unlucky. By the time populations expand again, that lineage may be stuck with a particular tendency, maybe a slightly extended juvenile brain growth period, or a particular pattern of connectivity between sensory and association areas. Later, when environments and cultures change, selection can work with that pattern, exaggerating some aspects, damping others, but the palette it draws from has already been reshaped. Or consider chromosomes themselves. Over deep time, chromosomes don't just sit there. They can fuse, break, invert sections, swap chunks in rearrangements that can affect how genes are inherited together. Our own species carries the legacy of such events. The fusion that produced our chromosome 2 from 2 ancestral ape chromosomes, or inversions that distinguish human, Neanderthal, and chimp lineages. Large-scale rearrangements can be risky. Carriers may suffer fertility problems if their chromosomes don't pair cleanly in meiosis. That tends to keep such variants rare when populations are big and well mixed. Any rearrangement that seriously compromises fertility gets weeded out. But in small, somewhat isolated groups, exactly the kind of structure implied by refugia and long-term low NE, odd chromosomal configurations can sneak through, especially if the group is already so small that mild fertility costs don't immediately doom it. Over hundreds of thousands of years, a rare rearrangement that begins as a liability in one little pocket can drift to high frequency there. If that pocket happens to be a core contributor to later Homo lineages, that once local quirk becomes a fixed feature of all its descendants. What started as a genetic accident in a dwindling refuge might, in the distant future, be interpreted as a hallmark difference between our lineage and our cousins. Another concept that becomes important here is lineage sorting. At any given stretch of DNA, ancestral populations can carry multiple diverging versions of a sequence. When lineages split, say an ancestral Homo population in Africa giving rise to groups that will eventually become modern humans, Neanderthals and Denisovans, those earlier variants don't line up neatly along species lines right away. For a long time, each descendant group can carry a messy mix of ancestral versions. Only over time, as some variants are lost by drift and others become fixed, do you get clean separation. One version common in one lineage, another in a different one. A long drawn-out bottleneck accelerates that process in strange ways. With so few ancestors contributing genes, the odds that all 3 or 4 ancestral variants at a given locus persist into the future shrink. Instead of a rich tangle of possible histories preserved in the genome, you get a pared-down set of lineages marching forward. When we compare modern human DNA to that of Neanderthals and Denisovans, some of the tidy-looking splits we see, this branch here, that branch there, may be the fossilized imprint of that deep pruning, rather than evidence of sudden adaptive breakthroughs. In other words, the bottleneck might have done a lot of silent editorial work on the genome, crossing out entire side plots, leaving just a handful of character lines for later evolution to elaborate. It's easy, standing in the present, to look back at that editing and want to assign it credit for the story that followed. To say, maybe the reason our species emerged with this particular combination of cognitive capacities, social tendencies and vulnerabilities is because of that ancient narrowing. Maybe it hardwired us for certain kinds of cooperation, or for longer childhoods, or for particular metabolic trade-offs. The honest answer is that we don't know, and we may never be able to know in detail. From a scientific standpoint, tying a specific biological outcome, a feature of our brain, our immune system, our growth, to a single demographic event 500,000 years long is extremely difficult. Too much has happened since. Our lineage experienced later population expansions and contractions, admixture with other hominins, adaptations to shifting diets and climates, cultural revolutions that change the selective landscape far more rapidly than genes typically move. Any one trait you point to today sits atop a mountain of interacting causes. Even if the bottleneck did shape which variants were available early on, later selection, chance, and culture all piled new layers on top. What we can say, with some confidence, is more general. A long period of low effective population size intensifies drift. It reduces overall genetic diversity and can allow odd combinations of genes, and even chromosome structures, that might have remained fringe in a large population to become standard. It can speed up the sorting of ancestral lineages, making some branches of our genetic tree seem cleaner and more distinct than they really were on the ground. It can leave us with a genome that is, in some places, surprisingly uniform and in others, strangely patchy. Beyond that, any attempt to draw a direct line from roughly 1,000 breeding ancestors for a long time to modern humans think and feel like this drifts into speculation. Still, the possibility that our deep vulnerability, our near erasure, helped sculpt what later became our strengths is hard to ignore. It suggests that the same processes that almost erased our lineage may also have quietly set boundary conditions on what humanity could become, and that circles back to the puzzle at the center of this story. If this bottleneck was so long, so influential, why did it happen at all? Why did our lineage squeeze through such a narrow gap when other mammals facing many of the same external pressures did not? And why, with all our tools and models, is science still struggling to say exactly what pushed us to the brink? Part of the answer is baked into that last question, with all our tools and models, because those tools are powerful, but they're also slippery. Start with time itself. To say that a bottleneck happened between about 930,000 and 813,000 years ago sounds reassuringly precise, but that range comes from models that have to translate patterns in DNA, patterns that accumulate over generations, into dates on a calendar. To do that, you need to know, or at least assume, how fast mutations pile up. You need to assume how often populations split, mix, or stay separated. You need to decide how many hidden demographic events you'll even allow the model to consider. Change those assumptions and the shape of the curve, the rise and fall of population size over time, can bend in different ways without being obviously wrong. Even the mutation rate isn't a single solid number. Different studies, using different methods, pedigree studies of living families, comparisons between species with fossil-based split times, ancient DNA calibration, sometimes give slightly different answers. Over a few generations, those differences barely matter. Stretch them over 1,000,000 years, and they can slide your bottleneck forward or backward by tens or hundreds of thousands of years. So when we say, 930 to 813,000 years ago. What we really mean is, given this set of assumptions and this way of translating genetic patterns into years, this is the window that fits best. Another team, using a slightly different clock or different demographic model, can nudge that window around without anyone necessarily making a mistake. Then there's the way those models simplify reality. To make the math tractable, they often treat the ancestral population as if it were a single well-mixed group that expands and contracts smoothly over time, or they divide it into a few big chunks that exchange migrants at tidy, predictable rates. The real world of early Homo almost certainly wasn't like that. It was probably a patchwork, some groups isolated for long stretches, others linked by rare but consequential movements, some pockets growing while others shrank or died out. That kind of structure can make a population look, in genetic hindsight, much smaller than the head count actually was. It can also produce signals, extended runs of shared DNA, odd patterns of relatedness, that resemble a global bottleneck, even if the crisis was more local and staggered. When you feed that messy reality into a model that expects smooth curves, the model does its best. It explains the patterns by bending the population size line down, holding it low, then letting it rise again. It may not be entirely wrong. The world really might have been thinly populated. But how thin, where, and for how long? That's where the interpretation starts to wobble. And then there's the ground truth problem. We have genomes. We have climate curves derived from things like oxygen isotopes in ocean sediments and tiny bubbles trapped in ice cores. We have scattered fossils and stone tools. But these data sets don't line up on a neat, shared timeline like layers in a perfectly preserved cake. They're more like pages torn from different books, overlapping in places, missing whole chapters in others. Genomes are global and continuous in time, but they're abstract. They tell us what must have happened to ancestry, broad rises and falls in effective population size, splits and merges, but they don't name causes. A dip in NE could be the echo of an ice age, or of a small number of refugia, or of a cultural innovation that let one subpopulation take over while others dwindled. The genome doesn't label the curve with famine here or volcano there. Fossils, when we have them, are concrete. An actual bone, an actual skull with a date attached. But the record for this window is painfully thin and unevenly distributed. Huge parts of Africa and Eurasia have never been systematically surveyed, or their sediments have been eroded, buried, or are inaccessible. The fossils we do have don't line up in neat time series that show a clear crash in numbers. Instead, we see gaps, no fossils here for 200,000 years, a few over there, that could mean no one lived here, or could just mean nothing preserved and we haven't found it yet. Climate proxies, meanwhile, are high resolution but blunt. They tell us that around this time, Earth swung into a regime of colder, longer, more severe glacial cycles. They show shifts in sea level, in dust deposition, in monsoon strength. They whisper that the world got harsher and more variable. But they don't single out humans. Many other species weathered those same swings without leaving such a dramatic genetic pinch in their descendants. Put these together, and you often get slightly different stories. Genetic models might say, long-standing, low-effective population size, starting a bit before a known shift in glacial cycles. Climate data might say, big environmental restructuring, but spread out over hundreds of thousands of years with regional differences. The fossil record might say, some hominin presence persists, with no obvious gap large enough to scream near extinction. Those small sample sizes make any strong claim risky. And then there are the local records, lake cores, cave deposits, volcanic ash layers, that hint at droughts, eruptions, and ecosystem churn in specific places that could have hammered particular hominin groups. Those can explain regional bottlenecks, but tying them cleanly to a global genetic signal requires exquisite dating that we often don't have. Even when dates overlap, causation is slippery. If a genetic bottleneck signal overlaps with a period of climate instability, is that enough to blame the climate? Or did climate change simply interact with other factors? Competition from other hominins or carnivores, shifting prey availability, disease outbreaks, social fragmentation, to deepen an already emerging trough. This is where the idea of multiple modest stresses becomes important. Science tends to look for big, clean causes: the asteroid that killed the dinosaurs, the super eruption, the single cataclysm. And those things matter. But in demography, you can also get a large, long-lasting effect from many smaller pressures piling up. Imagine early Homo living in a patchy landscape where good habitats are shrinking and moving. A prolonged cool spell doesn't kill them outright, but it pushes prey animals into new ranges and turns once reliable water sources seasonal. Competition with other hominin species cuts into hunting territories. Predators follow the same shrinking resources, increasing encounters. A disease emerges in one clustered group and burns through, leaving survivors wary of contact with outsiders. None of these alone has to drop the global census to a few thousand, but each shrinks, fragments, or isolates some subpopulation. Over centuries and millennia, the network of connections thins, migration routes close, gene flow slows, effective population size, the thing the genome is most sensitive to, drops, even if the raw head count in scattered refuges stays higher. From the vantage point of the genome, all of that gradual fragmentation and selective local misery can compress into a single smooth depression, the bottleneck curve. From the vantage point of fossils and tools, it might look like continuity with regional shuffling. From the vantage point of climate proxies, it's just one more stretch of a world in flux. So what would it take to do better than we're not sure? For one, more genomes, especially from parts of Africa that have been underrepresented in genetic studies. Different lineages within Africa may carry slightly different echoes of that deep bottleneck. Comparing them at higher resolution could help us see whether the bottleneck was truly global, across ancestral Homo, or stronger in some branches than others, hinting at where refugia were and how connected they stayed. Ancient DNA from this time period, if we can ever get it, would be revolutionary. Right now, ancient DNA barely reaches back a few 100,000 years in cold regions, and conditions in much of Africa are brutal for DNA preservation. But even partial sequences from slightly younger or slightly older hominins in key regions could anchor our mutation rates and demographic models more securely. Better calibration of our clocks also matters. That means tighter fossil dates, more reliable correlations between genetic splits and well-dated geological or climatic events, and improved methods for modelling population structure rather than smoothing it away. As statistical tools get better at handling messy, structured populations, some of what looks like a single long bottleneck might resolve into a more nuanced pattern. On the ground, new archaeological and fossil sites, especially in currently undersampled regions, could show whether there were expansive, thriving hominin populations in places our models currently treat as empty, or whether the thinness suggested by genetics is mirrored in actual occupation patterns. High-resolution local climate records tied to those sites could show whether particular environmental crunches line up with local population shifts. Even then, we may never get a single headline answer. Not the super volcano that almost wiped us out, but something more like a long period in which climate volatility, ecological change and social fragmentation conspired to keep our ancestors thin on the ground. And that, in a way, is the point. The story of this bottleneck isn't a failure of science to find the one true cause. It's a live, tangled research frontier where different lines of evidence are still being woven together. Every new genome, every redated fossil, every refined climate curve has the potential to slightly redraw the shape of that narrow passage. We're used to thinking of human origins as a finished tale, a sequence of key events, neatly labelled and dated. In reality, the period when our numbers may have dwindled to something like 1,280 breeding individuals is more like a half-erased section of a manuscript that researchers are painstakingly reconstructing from overlap, smudges and marginal notes. The mystery isn't a gap waiting passively to be filled. It's an active puzzle, one that keeps reshaping itself as new pieces fall into place. And our ability to ask better questions about it, to test whether this bottleneck was as deep, as long, and as consequential as it looks right now, is still growing. Which raises one more question as we turn back toward the present. What does it actually mean in lived experience to exist as a lineage passing through such a narrow corridor? To imagine that, we have to pull those threads together, not into a single tidy picture, because the evidence won't let us, but into a tapestry of overlapping possibilities, a world where several modest blows landing over a long stretch of time combine to press our lineage into that narrow channel. One part of that world is almost certainly climate, not a single glacial switch flipped overnight, but a deepening rhythm of cold and warmth that stretched and squeezed habitats. Forests retreating, then creeping back, grasslands shifting their borders by hundreds of kilometers, rivers changing course, coastlines walking tens of meters up and down with the sea. If you were one small band of early Homo, that meant occasional good years in a favoured valley where water still pooled and game congregated, but it also meant long, lean stretches, familiar migration paths turning into dead ends, grazing herds failing to show up where they always had, plants fruiting at odd times, or not at all. Layer onto that population structure, a patchy, fragmented human world. Instead of a single, continent-spanning population smoothly rising and falling together, picture scattered refuges, a group clinging to a lakeshore that shrinks and grows with the seasons, another tracking herds across a high plateau that's slowly getting colder, a third huddled in a coastal strip that widens during glacial lows and drowns during warmer highs. Between them, stretches of land that are sometimes passable, sometimes not. Dangerous corridors patrolled by predators or rival hominins, dry basins that only become stepping stones after rare, unpredictable rains. Contact is possible, but not guaranteed. Some years stranger appear on the horizon and trade, mate, share stories by a fire. Other years, no one comes. These songs and tool tricks and genetic lineages in one pocket of the map drift further from those in another. That patchwork alone can shrink effective population size without requiring the census to collapse to a few villages worth of people. Genes spread more slowly. Local crashes hit harder because there's less backup, fewer migrants wandering in to replenish what's lost. Then add competition. Not necessarily a dramatic war of extermination, but pressure at the margins. Other hominin species vying for the same watering holes, the same herds, the same patches of edible roots. Carnivores that have learned your patterns, that know your favourite ambush sites because they use them too. Hyenas and lions and saber-toothed cats whose own survival is tied to the same dwindling prey. Every carcass you claim is one they don't get. Every carcass they drag off is one your children go hungry without. In that kind of world, a harsh season can do more than thin the gazelles. It can stress your neighbours too, pushing them closer to your ranges. It can turn low-level competition into explosive conflict, or at least into a constant low hum of risk. Disease could have joined the chorus as well. We don't have direct evidence for specific pathogens from that time, but in small, semi-isolated groups, even a modestly contagious illness can have outsized effects. A fever that would be survivable with rest and food becomes lethal when there's no buffer, no granaries, no distant kin to help hunt, no medicine beyond what a particular band's knowledge can provide. Maybe one refuge gets hit harder than another. Maybe a group that's specialized in one kind of prey finds itself in trouble when that animal shifts its range. Maybe a volcanic ash fall in one valley poisons water and plants just enough to force everyone to leave, scattering them into territories already claimed. None of these need to be global catastrophes. Each can be regional, local, even episodic. But over tens of thousands of years, they chip away at connectivity, at abundance, at security. They turn what might have been a broad, robust network of early humans into a fragile webbed set of threads that can fray and snap. From our vantage point, we glimpse the sum of those processes as a deep contraction in ancestry, a signal compatible with our lineage being, for a long time, thin on the ground. That, the existence of a demographic pinch, a narrowing of lineages, is the part of the story that looks most robust right now. Multiple genetic methods, using different assumptions, keep pointing towards some kind of prolonged low in effective population size in this window of deep time. The specifics, though, are where the sand still shifts under our feet. Was the lowest point closer to a few thousand individuals, or were there more bodies spread out in small disconnected pockets whose genes simply didn't all make it through to us? Did the crunch coincide with a particular climatic shift, or did it stretch across several? Were certain regions, parts of Africa's interior maybe, or coastal corridors, relatively buffered, while others winked out entirely? We don't yet know how many of the people who lived through that era left no genetic trace in modern humans because their lineages ended in dead ends. Daughters whose children had no children, sons whose descendants were absorbed into neighbouring groups that later vanished. We don't know whether a key cultural or biological innovation, control of fire, more complex social structures, more flexible foraging, was already present before the bottleneck and helped us endure it, or emerged as a response to it. And we don't know the true head count at the narrowest point. The much-cited figure of around 1,280 breeding individuals is a model-based estimate, not a census. It's a way of saying, if we compress all this complex structured history into a single number that would have the same genetic effect, this is what we get. The reality on the ground could have been a mosaic, a few relatively stable pockets of hundreds of people each, with long empty stretches between, other regions flickering on and off as short-lived settlements rose and fell. Still, even with those uncertainties, there's a stark human-scale implication. At some point in that long, dim stretch of prehistory, the chain of ancestors that leads to you and to everyone you've ever met ran through a dangerously narrow corridor, not in a single generation, not in a single village on a single night, but across millennia in which the margin for error was thin. Every successful hunt, every avoided predator, every risky crossing to a new valley, every choice to share food rather than hoard it, every birth that survived and every child that learned enough to keep going, all of those small, ordinary triumphs added up. Survival, in that context, is not just strength. It's chance layered onto resilience, luck braided with learning, a storm that veers away instead of hitting a shelter, a drought that breaks one season earlier than it might have, a young wanderer who stumbles into a new group and brings genes, stories, techniques that make both bands more robust. The fact that we're here means that, against a background of grinding pressure, enough of those small favourable outcomes stacked in our ancestors' favour, not because they were destined to win, but because they were capable of adapting, and because, for reasons we still don't fully understand, the world didn't quite push them over the edge. When you look around at modern humanity, billions of us covering almost every habitable niche, it's easy to forget that our story includes a long chapter where we were rare, vulnerable, and one bad run of centuries away from disappearing. Remembering that doesn't just satisfy curiosity about the past, it reframes the present. It reminds us how contingent our existence is and how intertwined survival is with environment, with cooperation, with the unpredictable roll of the dice. If you've stayed with this journey all the way back through 1,000,000 years of uncertainty and difficult lives, take a moment to let that sink in. Every person you know, every language, every idea, every song exists because a thin thread of ancestors made it through that narrowest passage. If you found this deep dive into our fragile origins thought-provoking, it really helps the channel if you tap like, subscribe, and share this with someone who loves big, messy scientific mysteries. And I'd love to hear from you. Drop a comment telling me where in the world you're listening from and what this story makes you think about when you imagine our species' future.